Okay, welcome everybody to this one-day webinar on drug targets in cardiovascular disease. I'm Professor Amrita Ahluwalia. I'm a Professor of Vascular Pharmacology at the Barts and London Faculty of Medicine and Dentistry at Queen Mary University of London. I'm also the Chair of the the Basic and Translational section of the International Union for Basic and Clinical Pharmacology. We're hosting this one-day webinar. Essentially, this webinar came about from a conversation between myself, representing IUPHAR, and two colleagues, Professors Tom Gucic and Professor Pasquale Maffia. Both Tom and Pasquale have prominent roles, editor-in-chief and deputy, respectively, of the Cardiovascular Research Journal, owned by the European Society of Cardiology. And a few years back, over a conversation, we felt that there was a major opportunity to take advantage of a fantastic open database that was established by the BPS, the British Pharmacological Society, and IUPHAR, the Guide to Pharmacology. And we were discussing about the opportunities for cardiovascular researchers, basic and clinical, to access that database. And together with Jamie Davis and Steve Alexander, the two senior leads of the Guide to Pharmacology, we decided that there was an opportunity for us potentially to work with the Guide to Pharmacology leads to enable utility of the database for cardiovascular researchers, both discovery scientists and clinical. So I'm going to talk a little bit, you're going to hear a little bit more about the Guide to Pharmacology in just a moment, but just to say that we have a very packed schedule. What we are going to do for the first part of the meeting before lunch is that we're going to run through a number of presentations that you can see in the program. And at the end of those presentations, just before lunch, we will have a panel discussion that is led by Professor Pasquale Maffia. So we're very keen to have your thoughts, your queries posted within the chat. We will do our very best to pose any questions that you might have, all of the attendees, to the speakers. After the lunch break, we're actually, again, as I said, we have a very packed afternoon. We will endeavour to ask your questions of speakers after their talks immediately. But if we don't have time, please don't worry. What we will do is we will collate all of your questions and we will send answers to all of those who've registered for the meeting post the meeting, together with the recording of this event. Okay, so without further ado, I shall introduce the first speaker. So the first session this morning is really giving an idea of the Guide to Pharmacology, its history, its structure, and its potential utility. And the first speaker that we have is Professor Steve Alexander. Steve is the chair of the nomenclature committee for the International Union of Basic and Clinical Pharmacology. He's also a professor of pharmacology at Nottingham University. And he has played a very prominent role, an important role in unifying agreement across the globe with the support of international leading researchers on targets and ligands. And he's going to give us a little bit of the history and the detail behind the Guide to Pharmacology first. So, Steve.
Thank you, Amrita. Good morning, everybody. For those of you in Europe, for those of you slightly eastwards, good afternoon. What I'm going to do is give a bit of a background about where the information comes from that we communicate through Guide to Pharmacology and the mechanisms whereby we maintain it as a kind of rich source. So I have this title of Communicating the Rational and Systematic Nomenclature in Pharmacology and Therapeutics. And that comes from about 30 years of the Nomenclature and Standards Committee of IUPHAR, as Amrita has described. So just going to talk about two areas, what it is that we do at NCIUPHAR, and then how we communicate this, because of course it's not just about the science, it's about good communication too. So basically how we do what we're doing at NCIUPHAR is based around a huge network of scientists globally, over 500 scientists at last count, in at least 106 subcommittees. So NCIUPHAR was started as the International Union of Basic and Clinical Pharmacology's Committee on Receptor Nomenclature and Drug Classification. And at the time in 1992, when Paul Van Hoot set it up, the focus was definitely on receptors as the primary target. But of course, since then, we've expanded hugely into every other area of drug targets. So the primary objectives, and I've highlighted a couple of them here, were to look at guidelines for the nomenclature and classification of human biological targets, and particularly to develop this authoritative, freely available global online resource, the Guide to Pharmacology, which is present at that URL. So the way in which NCIUPHAR works is that there is a core group of mostly academic and a few industrial scientists who review and revise nomenclature based on relationships with multiple subcommittees. And we've kind of organized into a traditional pharmacological grouping that would identify drug targets as receptors, ion channels, transporters, and enzymes. And inevitably, as we try and pigeonhole biology, what it does is it pushes back. And so we have this group of atypicals which don't readily fit into those other classes. And within each of those areas, we've got subclasses. Necessarily, the complexity of receptors means that we've got at least 4 groupings. Transporters and ion channels are also subclassified. And you'll see that biology again gives us that wrinkle, if you like, of receptors which are also ion channels. We have enzymes which are also receptors as well. But the aim is to have this hierarchy that allows a logical and systematic view of all the drug targets. And so within these, there are a number of subcommittees which are usually looking after families of targets together, which evaluates the current nomenclature and work on elements of the pharmacology that gets reported back through NCIUPHAR. So these, at the moment, more than 500 scientists, and since the instigation of NCIUPHAR, that's well over 1,000 scientists have contributed to the information base. So the information base needs to be communicated, and we do this through 3 primary publication streams and the online database. So the hierarchy that we have is NCIUPHAR is a visible exponent of the International Union of Basic and Clinical Pharmacology because of these outputs. And it lies in and interacts with 2 major national pharmacological societies, the BPS, who are hosting today's webinar, and thank you for that, and the American Society for Pharmacology and Experimental Therapeutics. And the relationship we have with those 2 is because of the outputs that they have. The society journals, which are very high profile, the British Journal of Pharmacology and Pharmacological Reviews. And NCIUPHAR has been publishing reviews in both of those journals for some time. One of the things that makes the availability of the information free is the interaction with the British Pharmacological Society and the Guide to Pharmacology.org website. And from that, we take every 2 years a snapshot of the information. It's an enormous body of information we have there, as you'll see in a second. And so what we do is we take limited views of that and put it into the only paper output the British Journal of Pharmacology still has, which is the freely available concise Guide to Pharmacology. We've been able to do this largely because of educational grants from Servier, and that's been down to previous heads of the NCIUPHAR and the relationship with Servier, and that's been able to allow us to have those core meetings. And what we'll talk about later today as well in a second is the possibility of producing specialist portals for the pharmacological community, the Guide to Immunopharmacology and the Guide to Malaria Pharmacology. So the three major outputs that we have, the pharmacological reviews and the British Journal of Pharmacology and the Concise Guide to Pharmacology, I've just given you an impression of how these are used by the community, because I've just given you an indication of citations here. So on the left-hand side, you have pharmacological reviews, where we've published 112 reviews since 1992. And these have accumulated a really impressive number of citations, 46,000 or thereabouts. And in the centre, the British Journal of Pharmacology, which is a more recent collaboration, just over 10 years, where we've got 37 reviews. So whereas the pharmacological reviews output is mainly focused on nomenclature and establishing systematic and rational nomenclature, the BJP outputs are for many other aspects of pharmacology where there is an established nomenclature. And then on the right-hand side, what you can see is the Concise Guide to Pharmacology, which we previously called the Guide to Receptors and Channels. And this emanates from something that one of our speakers today, Steve Watson, instigated as the receptor nomenclature supplement for the Trends in Pharmacological Sciences. And then myself and John Peters took that over as an editorial role. And with Alistair Mathey, we took that to the British Journal of Pharmacology and started putting together the Guide to Receptors and Channels and now the Concise Guide to Pharmacology. And we've had 10 editions, and we're in the current cycle of producing the 2013, 2023, 2024 edition of the Concise Guide. So the online database is the way most people get access to the information. So it was instigated as IUPHAR DB in 20 years ago by Tony Harmer. And it was based in Edinburgh, because that's where Tony was based. And the information that IUPHAR had at that time was mainly focused on established targets, G protein coupled receptors, nuclear receptors, and ion channels. And as I became part of NC-IUPHAR, this expanded further into enzymes, transporters, and the atypicals. In 2011, we established the Guide to Pharmacology.org, which was described as a collaboration between IUPHAR and the BPS. And the aim there was to develop something which was a single entry point, a one-stop shop, as it was called, for information on pharmacological targets and their ligands. And what I'm going to do now is describe some of the Guide to Pharmacology content. Obviously, there's a huge amount there, and we can't cover it all, but I'll just cover some of the ways in and the major soundings, if you like. So this is the homepage, the IUPHAR BPS Guide to Pharmacology. It has the usual things that you'd expect from a database, online database, the browse function, the search function. We have news and hot topics that get updated often weekly, and then link out to other resources. We have the capacity for downloads in all sorts of formats. And of course, we have a Twitter location as Guide to Pharm. So just an example of how one might get into the information that's held on the database. If one were interested in G protein coupled receptors, then there's a long list of those GPCRs there. And if we pick on one, the 5-HT receptors, 5-hydroxytryptamine, serotonin receptors, there's a list accessible of all of those G protein coupled receptors that respond to 5-HT. And if we pick out one, the 5-HT1A, you can see that what we do is we develop a hierarchy which describes some of the genetic descriptions of that receptor, linking out to places like Ensemble, UniProt, the Human Genome Nomenclature Committee. We describe transduction, agonists, and selective agonists, and selective antagonists and labeled ligands. And what you see, this version is a condensed version. This is not all of the ligands. And one of the reasons we do that is to promote the idea of gold standard compounds. When we look at individual compounds, what we can do is get physical chemical properties, structures and the like. But when you click on those individual ligand pages, you can get a lot more detailed information, target interactions with other targets, for example, And one of the key things that we do is we promote this idea of the evidence base by linking directly through to the literature, usually PubMed, but occasionally to patents. And the content of the Guide to Pharmacology in the last release, the last quarter of last year, we're just about to release the next version. We include 3,000 plus drug targets, as you'd expect, the majority of enzymes, a large number of transporters, G-protein coupled receptors, catalytic receptors, and so on. And around about 11,000 plus ligands. So the majority are synthetic ligands, and one of the things I've highlighted here is the number of approved drugs. And one of the things that we obviously try and maintain is the currency of our list of licensed drugs, and that obviously increases year on year. And then we go down through things like natural products in the back, and label ligands and a small number of inorganics. So one of the things we're trying to do with these databases is not just list things, but to add value. And one of the ways in which we add value is we use our expert scientists to recommend gold standard tool compounds, which can allow the identification of drug targets in complex situations. And clearly, one of the things that pharmacology allows is quantitation. And so we're very keen on the quantitative side of pharmacology. We report over 19,000 curated binding constants, and we have that major link to the literature, over 40,000 references. We maintain the currency because we have this quarterly release cycle that allows the most recent updates to be available for people to look at. And just to give you an idea of the kind of usage of the database, on the left-hand side, what you'll see is monthly statistics in the brackets underneath the previous 12 months from leading up to 2022. So for the last 12 months, we had around about 50,000 sessions per month and over 36,000 users per month with almost 150,000 page views. So not only are we presenting information that is curated and expert viewed, but clearly it's being consumed by our target audience. So what I'm going to do now is hand over to Jamie Davies, who's going to tell you about some of the ways in which we catered for specialist pharmacological populations over the last 10 years.
Okay, so Steve's just introduced and explained the rationale behind the database and how it works, and has shown you some pictures from it. One of the things that we're very conscious of is that although what we're developing is a database which is primarily for pharmacologists, a lot of people who can get real use out of it and use it to promote their research and ideas wouldn't describe themselves primarily as pharmacologists. They describe themselves primarily as specialists in other subjects who happen to need pharmacology. And pharmacology, it's one of those things that well, all scientific subjects really, different specialties see them in different ways. It's not really like the men and the elephant in that everybody does have some idea of what it all looks like, but they've got their own preferred views. And where we have a user community that wants to engage with us like this, we have some experience in producing different front ends to our database to suit different communities. So just to be clear, the actual database, the content, is exactly the same under the hood. Behind these front ends that people see when they come in from the web, there's the same database, the same information. It's just that people may want to access it different ways. An example of this was a portal that was constructed for the immunological community, and particularly for people who would consider themselves, if you said, "What are you?" academically, they'd say, "I'm an immunologist," but they still had reasons to be using drugs as tools, or to be trying to develop drug approaches to control of inflammation, or to boost immune reactions, or whatever it would be. And while pharmacologists tend to think in terms of ligands and targets and binding and that sort of thing, which is why the main page that you come into when you look at Guide to Pharmacology puts those things front and center, immunologists tend to think in different ways. So a typical immunologist would think in terms of cellular processes, such as antigen presentation or B cell activation or that sort of thing. And will also think in terms of cell types. They'll be interested in, dendritic cells, or they'll be interested in T cells or mast cells. So for that reason, we've generated a different front end, which is called the Guide to Immunopharmacology. And here we've got these things like processes and cell, sorry, I don't quite know what happened then. Processes and cell types. These are just the boxes you saw on the last slide, but I've made them a little bit larger, so that people can come in and click on something like immune regulation or click on natural killer cells and see the pharmacological information selected, coming back prioritized to match that kind of thing. So for example, if they clicked on inflammation, then they'd get to a submenu, which is organized around gene ontology terms. And these are all aspects of inflammation, which they can click on to then get targets and ligands, which are closely associated with each of those gene ontology terms, so each of those sub-processes. And this was all designed in collaboration with a bunch of immunologists who gave up their time to help us, which is very kind of them, so that we know that this is actually, it conforms very closely to the way that immunologists think about things. And we ran test versions of the database, or the interface. We had exercises of people coming up and using them just to kind of tweak and to make sure that we had captured this properly. A second example of this was made in collaboration with the malaria community, particularly the Medicines of Malaria venture. And that's the Guide to Malaria Pharmacology. And that was a little bit of a new departure for us because, as Steve said, most of our targets have been human up until then. But obviously, for a type of pharmacology which is treating a parasite, some of the targets are non-human, they're targets within the parasite. So again, this different front end is optimized for people in that community and was optimized in collaboration with people in that community. And they still think of targets and ligands, but they'll also think, for example, of parasite life cycle stages, because the malaria parasite has got several different stages to its life cycle, and they're amenable to different treatments, and they have different pathological consequences. And also, there are different species of parasite that cause malaria. It's not a single disease. There are several different kind of subspecies of parasite, and they result in different types of malaria. Here's an example, if somebody clicked on the life cycle part, then they get a little bit of background, because all of these sites are also educational. So though they're designed primarily for professionals, there's always material behind which can be educational. And of course, professionals from an allied field will still need the educational material. I know I certainly do, whenever I'm exploring these things. And then from here, it's possible to click on the life cycle stage, for example, and then clicking on that will reveal targets and ligands that are active against that life cycle. And this is an example. So if you hit on one particular life cycle stage, here are a lot of interactions between targets and ligands, which can be then clicked on and open up to see their structure and various other attributes to them, just as in the main pharmacology database, because this is the main pharmacology database behind. This is just a new way in. This is an example of what happens when people follow one species of mosquito rather than following a life cycle stage. And I'm sure you get the idea. We're currently building a version of this which is optimized for bacteria, and that's being done in collaboration with a global antibiotic research and development partnership, GARD-P. We've been working with them for a while on their antibiotic database, and that now links very strongly to the Guide to Pharmacology database. So where there's solid pharmacology behind antibiotics, there isn't always. Some antibiotics still don't have defined targets, but where there is, then all of the information is available through our main database. And we're now going to be building a new front end so that people in the microbiological community can come to the database and see a front end that's optimized for the sorts of ways they think and the kind of questions they want to ask. And we've been showing you this. We've been showing you the whole database because if you don't already know it, we really recommend, it's a very useful resource. And we're also showing you this because it would be wonderful to be able to do this kind of thing for the cardiovascular community as well, so that there would be a way of that community having a portal that's absolutely optimized to your needs. Okay, thanks very much.
Okay, thank you.
relinquish the screen.
Thank you both, Steve and Jamie. So I think with that, we have a really good idea of the potential of the Guide to Pharmacology database. The aspiration that this webinar has been constructed around is that we determine whether the cardiovascular community, both discovery, basic science, and the clinical research community, could benefit, could get value out of a portal that was actually specifically targeted to enable access to that community. What's very clear is that having specialized focused areas that enter into the Guide to Pharmacology has opened up the amazing wealth of data that sits within that database to communities that actually need that data. Our aspiration is that we create potentially something that will support researchers and research in providing information regarding the best tools for targets, but also, of course, in drug development, developing novel drugs based upon the information that is exists within the Guide to Pharmacology. But also, as Jamie mentioned, it's an invaluable educational tool. And it's clear that there is an opportunity here that the cardiovascular research community might benefit from. The first step in realizing that aspiration essentially was to garner support from key organizations that would help us to recruit a number of international leaders in some of the most exciting and hot topic areas currently within the cardiovascular research field, but then also to use that opportunity to garner opinion from the cardiovascular community regarding how the portal might be structured, whether or not there would be benefit for the community, and who might be involved. So all of those of you who are here, who have registered for the meeting, we're very keen to hear your opinions. We wouldn't have been able to set up this webinar without the support of two key organisations. The British Heart Foundation have been enormously generous in supporting us and funding us to set up this webinar to test the environment, to determine whether there is an appetite for creating this portal, and to hear from some of our international leaders across the piece. And also the British Pharmacological Society, who've supported us with all of the logistics of this meeting. So I think without further ado, what we should do is move to the science. We're going to hear from, as I said, a number of international leaders in highlighting a variety of different targets that are relevant for drug development. And within their talks, hopefully we'll be highlighting some of the key issues that a cardiovascular portal for the Guide to Pharmacology might help and support in dealing with those issues. Anyway, so I'm now going to hand over to Professor Pasquale Maffia, who's going to chair the next session. Pasquale.
Thank you, Amrita. Thank you also to Stephen and James for giving us a very broad overview of the nomenclature committee, the work done by the Guide to Pharmacology, and to Amrita for explaining our ambition to create the Guide to Cardiovascular Pharmacology. If you don't know me, my name is Pasquale Maffia. I'm a professor of cardiovascular immunology at the University of Glasgow. I'm also the chair of the Immunopharmacology Committee in IUPHAR and deputy editor of Cardiovascular Research. We can start with the first session. We will have a series of 8 talks from opinion leaders in the field. The first session is dedicated to molecular pathways in cardiovascular disease. Can I remember everyone that if you have any question, you can please post your questions in the chat, and then I will convey your question during the discussion at the end of the session to the speakers. Forward to the next speaker, which is Professor Ziad Mallat from Cambridge. Ziad has been pioneering the understanding and study of immune mechanism in cardiovascular disease in the last 20 years, in particular with the focus on atherosclerosis, and now is translating this knowledge into the clinical scenario. So Ziad, thanks again for accepting the invitation, and we are looking forward to your talk.
Yeah, thank you, Pasquale, for the invitation and the introduction. Can you see my slides? Yeah.
Yes, we can.
Okay, great. So I'll be talking from totally different perspectives. You all know that I'm not a pharmacologist. I'm a cardiovascular scientist with interest in the role of the immune system in cardiovascular disease. But I will show you how during our studies, how we were facing in some of the development of therapeutics in cardiovascular disease, we were facing some aspects of immunopharmacology and how knowledge of this can improve the way we are choosing the right targets or the right agents to target these targets. So I will start with the general overview of the immune system and its role in atherosclerosis and cardiovascular disease. And we'll go through two different areas of therapeutic targetings in cardiovascular disease using manipulation of the immune system. So this slide is really to show you that the immune system has intricate relationships with the vascular wall. Already in a healthy vascular wall, there is an important interaction between immune cells, whether in the intima or in the adventitia, with the vascular wall. You can find at baselines in a healthy vessel, interaction of circulating monocytes with the artery wall, with some homeostatic functions. We know that there are intimal macrophages are already sitting there and playing a role in the homeostasis of the vascular wall. There are adventitial macrophages also playing an important role in the remodeling and the response of the vessel wall to hemodynamic stress. There are T cells and B cells that are sitting in the perivascular adipose tissue and that also ensure vascular homeostasis. And how we see the development of atherosclerosis now is really a breakdown of this homeostasis of the immune vascular interactions that go on to induce vascular inflammation and exaggerated recruitment and activation of immune cells and vascular cells, leading to the development of and the progression of the disease. Of course, I will not go through all of these, and you can imagine a lot of immunocardiovascular pharmacology here. And this is not only true in the vascular wall. I mean, the heart itself is also in healthy situation, infiltrated with immune cells, whether within the myocardium itself. We can find macrophages, dendritic cells, T cells, Tregs, whether in the pericardium with the pericardiac adipose tissue, a lot of immune infiltration at baseline that are ensuring cardiovascular homeostasis. And again, it is this disruption of immunocardiac interaction and homeostasis that is important and leading in the progression of the disease and activation of overactivation of the immune system and impacting cardiac remodeling, for example, here, post-ischemic injury. So, as you know, it's a huge area now in development, targeting the immune system in cardiovascular disease. And you know a lot about targeting some of these pathways that have been tested in the clinic. The IL-1 pathway, notably with canakinumab, colchicine and other agents here, anakinra, etc. And then you know about the proof of concept that these therapies have provided in cardiovascular disease, showing, for example, that targeting the IL-1 pathway is protective in cardiovascular disease. However, it comes with side effects that we should try to prevent. A lot of development in this area with targeting the IL-1 alpha pathway or targeting the IL-6 pathway. And these are all in the same IL-1, IL-6 pathway that is being now in the clinic. I will talk to you about other areas that we are now developing, mostly targeting the regulatory T cell immune response in cardiovascular disease and targeting the B cell response, with many other areas in development. So let's start with the B cells. A long time ago, we found something unexpected, is that some types of B cells called mature B2 cell types, coming from the follicular part of the B cell immune response, are pro-atherogenic, and that their depletion reduces the development of atherosclerosis in mice. We also showed later on that the same type of B cells is activated following ischemic injury to the heart, and this leads to the activation of this B cell phenotype and an interaction with other immune cells, for example, here through the production of chemokines that recruit inflammatory monocytes to the ischemic heart and promotes abnormal cardiac remodeling, increased infarction size, and reduction in ejection fraction. So here, where I'm talking about from a naive perspective. So here we identified that, for example, the CD20 expressing B cells are critical in this process, that they are activated by this B cell activating factor through a BAFF receptor. So you could try to target this pathway either by blocking and interfering with the BAFF pathway, or directly interfering with CD20 expression on the B cells and its role in B cell biology. So if you look, and when I discussed with Pasquale Maffia about this guide to pharmacology, I wanted to know, what would you know about consulting this guide. So in terms of BAFF, for example, if you go to immunoligands, you can find that BAFF has different receptors, BAFF receptors, BCMA, TACI. You can find that these receptors are not highly specific for B cells, and they can be expressed on immune cells. So in this case, it would not be suitable for us. You can find that you can target BAFF with different existing monoclonal antibodies that are in clinical use. You can also find that targeting this BAFF pathway does not lead to immediate change in the function of B cells, because in fact these therapies have been developed also for chronic disease, and it was not the aim of targeting acute events. So we can learn from this that these monoclonal antibodies targeting BAFF would not be really useful in an acute setting. You can go to CD20 and you can see all the monoclonal antibodies that are in use, in clinical use, to target this pathway. And you can find more information about these and their use in the clinic, their side effects, etc. And on this, we choose rituximab in order to see whether, because rituximab is very specific for CD20 on B cells, it's a humanized antibody against anti-CD20, depletes B cells quite rapidly, and this could be an interesting molecule to test in patients with acute coronary syndrome. So we went on with the first study to test the safety of rituximab in patients with ST elevation acute myocardial infarction. knowing that currently rituximab is contraindicated in patients with acute myocardial infarction. So we had to go through a safety trial initially to ensure that it is safe in these acute setting, and to also look at its potential biological efficacy in the setting. So we tested this compounds in patients with acute ST elevation MI that have undergone successful revascularization, and with exclusion criteria that these patients have a stable disease after successful revascularization. And we wanted to test, in fact, different doses of rituximab. What you cannot find now in the literature is the impact of different doses of rituximab on the biology of B cells in vivo in humans, because most of the studies were conducted with this 1,000 milligram dose and in repetitive dosing. Here, our aim was to inject rituximab as a single infusion at the acute phase of MI, and therefore we wanted to test different doses of rituximab and see their safety and their impact on the B cells in this acute situation. And we wanted also to understand whether their impact on the B cells was very rapid or not, because you are in a situation of acute myocardial infarction and you would like to impact the disease process very acutely and very rapidly. And this information, again, is not in the literature. For example, we cannot find in the literature whether the impact on B cell depletion with rituximab is very rapid or not. So we chose to analyze very early time points, baselines versus 30 minutes during the injection, day 6 and 6 months. As you can see here, this is a new information showing that even very low doses of rituximab are very effective in clearing the circulating B cells very quickly at 30 minutes already after the start of the infusion in patients with acute myocardial infarction, and that the impact of different doses of rituximab is different on the recovery of B cells during the 6 months following the administration of rituximab. For example, with the 200 milligrams, we have a reconstitution of B cells of particular phenotype that we think might also be protective in the setting of post-MI. So based on this, we have shown that injection of these different doses of rituximab was safe at the acute phase of myocardial infarction, that even the lowest dose can deplete circulating B cells very quickly, and has different impact on the recovery of B cells after myocardial infarction. So based on this information, we moved to a phase 2b RITA-MI2 trial, which is a European multi-center clinical trial that is funded by Horizon Europe and is now ongoing. It has started, and in which we are testing 2 different doses of rituximab, the lowest dose, 200 milligrams, and the highest dose, 1,000 milligrams, at a single infusion versus placebo at the acute phase of myocardial infarction, with the aim to look at the impact of these doses of rituximab on the recovery of heart function, left ventricular ejection fraction as a primary endpoint measured at 6 months by cardiac magnetic resonance imaging, at 6 months after myocardial infarction. So this was important in terms of characterizing the impact of 2 different doses of rituximab, their impact on the acute depletion of B cells and the recovery of the B cells, and this is how we were able to choose these 2 different doses to move on to the phase 2b clinical trial. But also, rituximab has different biosimilars, and the questions came during this phase 2b clinical trial whether we will use only one version of rituximab, which is the initial version developed by Roche MabThera, or other biosimilars. And you can have a look at this immunopharmacology guides to see that all these biosimilars have been approved and have similar pharmacology to the MabThera antibody. And then we allow, during the SWITA-MI2 trial, the use of all these approved biosimilar of rituximab in the trial. Now, the second trial that we are, the second therapy that we are developing in cardiovascular disease is targeting regulatory T cells using interleukin-2. And this is again an important immunopharmacology. As you can see here from this slide, interleukin-2, recombinant interleukin-2 has been approved initially and is now only approved for treatment of cancer disease and specific types of metastatic renal cancer because of the role of IL-2 in activating effector T cells here in red in order to combat tumor cells and limit their development. But these studies in metastatic cancer failed, in fact, in showing important benefit. And one of the most important reasons for that is that IL-2 also activate regulatory T cells that are not suited to combat cancer diseases. So as you can see here, the interleukin-2 activate regulatory T cells, but it does so already at doses that are 1,000-fold lower than the doses that are required to activate effector T cells. So this is an important, interesting immunopharmacology here. And then based on this, people have now shifted interest in interleukin-2 in order to use very low doses of interleukin-2 in order to selectively activate regulatory T cells without touching on the effector T cells in patients with autoimmune disease in order to reestablish a balance, increase their Tregs and reestablish a balance in the immune response and help limit the overactivation of the immune system in these disease settings. And we wanted to apply this to cardiovascular disease in the case of acute coronary syndromes, because we knew that Tregs are important in the setting and that they are reduced at the acute phase of myocardial infarction and acute coronary syndromes, and promoting them would be beneficial. And we wanted to test different doses of IL-2 in this range of what we call low doses, because nothing really was known about how different low doses of interleukin-2 may behave in patients with acute coronary syndrome. So we launched this LILAC trial, repurposing this recombinant interleukin-2 in patients with coronary artery disease. And again, since interleukin-2 was and is still contraindicated in patients with acute coronary syndromes, we started with a part A of the trial with patients with stable ischemic heart disease and testing different doses of low-dose interleukin-2 in a dose escalation manner, starting with the lowest dose, ensuring safety and biological efficacy, and moving to the next dose. And we tested 5 different doses of low-dose interleukin-2 in these patients compared to placebo in a randomized manner. So this was daily subcutaneous injection of interleukin-2 for 5 days, after which we measure Tregs and T effector the day after the final injection of the 5 days. So this is at day 6, and 1 week after the last injection. And once we have finished this part A, we've been able to choose 2 different doses to move on for testing in patients with acute coronary syndromes in a part B study. So this is published in EJM Evidence, so you can have a look at the paper. It is safe. We have found no serious adverse events. Most of the adverse events were transient and related to injection site reactions. And in terms of biological efficacy, this is the efficacy of the different doses of interleukin-2 in patients with stable ischemic heart disease. And you can see dose-dependent effects using these low dose ranges of interleukin-2. And we moved on with these 1.5 and 2.5 million unit daily dose. And you can see that these 2 different doses substantially increase Tregs in these patients without touching on the T effector cells. However, you can, when looking at these diagrams here, you can find that still start to find some differences between these 1.5 and 2.5 doses. And we wanted to examine this in more detail before moving to phase 2b clinical studies and choosing the appropriate dose that we should use in the next step. So if you look here at the day 6 versus baseline differences in the different T cell subset, you can clearly see that the 1.5 and 2.5 dose have a very selective effects on Tregs at day 6 in comparison with the other cell subsets. However, 1 week after the last injection, the 1.5 dose in blue here still have some very selective effects on Tregs. However, the 2.5 million dose seems to have some spilling over to the other T cell subsets. So we wanted to analyze this in more detail, and we've conducted a study to do single cell RNA sequencing on the peripheral blood immune cells in these patients before and after the injection of these 2 different doses of IL-2 in patients with acute coronary syndromes. And this was really very helpful in understanding the biology and the immunopharmacology of IL-2 in patients with acute coronary syndromes. We analyzed more than 200,000 immune cells in the blood. You can see here that we confirm the impact of the 1.5 dose and 2.5 dose versus placebo on the Treg population. We've been worried a bit about the impact of IL-2 on NK cells, which are known to be to have cytotoxic effects that you don't want to see in patients with acute coronary syndromes. And this single cell analysis, in fact, allowed us to be reassured, because we have been able to see that this type of NK cells that are increase in response to IL-2 are CD16 negative NK cells that are in fact known to be immunosuppressive. We've done additional analysis on these immune cells, and as you can see here, in blue the 1.5 dose and in orange the 2.5 dose, comparison of Tregs with T effector memory or T central memory shows you immediately here that the Treg, that the 1.5 dose in blue was very specific to Treg modulation in comparison with the T effector and the T central memory modulation. And this is based on all of these data, we chose to move on with the 1.5 million dose for the IUPHAR trial, which is a phase 2b trial in patients with acute coronary syndromes, randomizing patients with acute coronary syndromes to either placebo or 1.5 million dose of interleukin-2, and for treatment for 8 weeks, and the endpoint will be assessing vascular inflammation by FDG PET-CT at the end of the trial. This has been completed now. The data is being analyzed, and I hope that in the next 2 or 3 months we will have the data published and released. So if you look at this, I mean, what could be the next step? For example, you can see that a lot of biologicals related to interleukin-2 are being developed. Interleukin-2 with improved half-life, for example. Other IL-2 biologics with improved Treg selectivity are being developed. So a lot of research in this area that can be harnessed in the future for the treatment not only of autoimmune diseases but also cardiovascular diseases. Looking back at our data, you can see here by analyzing single cell interactions in the immune cells, you can find that one pathway that the IL-2 has drastically modified is this CD28, CD86 T cell dendritic cell interaction pathways that you can see here, which was absent in the placebo. And interested in this, you can look at the immunopharmacology guidelines to see that, in fact, this pathway of CD28 is already an interesting pathway that is being targeted in other disease settings. An antibody, an agonistic antibody, failed because it has led to very important cytokine release syndrome, so that it was removed from further development. However, now there are other anti-CD28 antibodies that are being developed that prevent co-stimulation and cytokine release and could be also important in the cardiovascular disease setting. Now, I will finish by saying that even though this is an interesting information that we gather from the immunopharmacology guideline, I found that most of it is related to rheumatological diseases and cancer, and not really to cardiovascular disease. For example, if you look at colchicine in this immunopharmacology guideline, you cannot find any information about colchicine and its role in cardiovascular disease treatment. And we know now a lot about colchicine in cardiovascular disease, and with phase 3 trials showing efficacy of colchicine in treating patients with cardiovascular disease. So there will be an interesting development and interaction to improve this, and a cardiovascular pharmacology guide in order to integrate this information about the immunopharmacology to cardiovascular disease setting. Similarly, there is an anti-IL-6 ziltivekimab that is being developed in cardiovascular disease, but you can find no information about this here, despite the fact that you can see that it is mentioned that it's developed in cardiovascular disease settings. However, you can find no link to clinical trials currently in cardiovascular disease setting using this antibody. Similarly, you can find a lot of information about ligands in the immune system. For example, APRIL and BAFF that I talked about before, involved in immune modulation, but we know that these immune ligands and their receptors are also expressed not only in immune cells, but also in cardiovascular cells. And this information could be very interesting to have and provide in order to have more better information about what modulation of the system, how this modulation of the system can impact on cardiovascular disease. For example, we have recently published in collaboration with Christoph Bender on the role of this APRIL, this immune modulator, in cardiovascular disease settings, and we discovered that this APRIL cytokines or growth factor can also impact vascular biology directly without impacting the immune system. So an important link between immunology and cardiovascular biology here in order to be able to develop cardiovascular targeted therapies with the use of immunological information. I will stop here and will thank my collaborators in Cambridge, within my group, within Mena Patworthy group, collaboration elsewhere, and the clinical trialist with Joseph Cherian and Steve Hoole here in Cambridge on the low-dose IL-2 and rituximab trial. Thank you very much for your attention.
Yeah, thank you very much for your talk, which has been interesting and stimulating as usual. Also, thank you very much also for going through the guide and highlighting strengths and weaknesses. That is exactly the goal of today's meeting. And clearly, we need an interaction with cardiovascular people to make it stronger and better, and this is exactly what we want to develop. We have now around 30 minutes of discussion that will be chaired by myself and will be with the two speakers of the last two talks, but also with the previous speaker of the introductory section, which is Amrita, Steve, and Jamie, that will be acting as panelists. We have also several questions in chat, and I will go through all of them. Can I just start from the last point that was raised by Ziad, because I think this is an important point. So maybe, Jamie, do you want to give us an update of exactly how the guide is updated on a regular basis, and how the interaction with the cardiovascular community may make the process more robust?
Yes, so the way that we update the guide, there are two main ways. One is curator looking for new information, and the other one, which is an important one, is all of the subcommittees of IUPHAR, who are the people that Steve talked about earlier, who have, each committee being interested in a particular target family. So, for example, the GPCR family, or an ion channel or something like that. One of the problems we have, well, we have two problems with that. Frankly, one problem is limited curator time, but the other one is that there will always be communities within science, for example, the cardiovascular one, that are not brilliantly represented on all of the target subfamily committees, which means that while things like the molecular interactions of the subcommittees we capture pretty well, the existence of trials in a particular area of medicine we capture less well, particularly because the core pharmacology of the molecular interactions is our absolutely core work, whereas the way that things are being developed, we'd love to capture it all, but we need help from communities to do that. And that's one of the reasons we're really keen to interact with the cardiovascular community, because we'd like to get this information into the guide and to keep it fully up to date for that community.
Okay, perfect, thanks. Can I start with some question on the science? Ziad, I mean, your work is interesting as usual. I was surprised, for example, to know that you can see depletion of circulating B cells in 30 minutes following rituximab. This is pretty interesting. Do you have any data in the preclinical setting of what happens on B cells in the tissue, how fast they are depleted?
Yeah, this is very important. I mean, we were very surprised when looking at the literature not to find any good information about how fast are B cells depleted in human beings after rituximab. And I can understand why, because probably this information is available from the industry, but the industry has not published this because they developed this in clinical trials. But there, the development of these antibodies, for example, rituximab, was aimed at chronic diseases, not acute diseases. So it's not very important to know whether rituximab is able to deplete B cells very quickly. And this is why this was not of interest probably to the community. And then we wanted to have this information and see whether in a very acute setting, you can have a very quick depletion of B cells, and this is why we chose to analyze the 30 minutes time points, and we can see effectively very rapid clearance of these B cells from the circulation. Now, you're right. What we don't know is whether the tissue resident B cells, in the lymph nodes or in the peripheral tissues of the spleen are depleted as quickly as possible. We think that this is happening because there is no big. When we look at other time points, the recirculation is very, very low. They recirculate these B cells, but we don't have any additional information from humans. In mice, we know that this is happening. But you are correct. In human beings, we don't know a lot about tissue resident B cells, and I think that there is a lot of opportunities here to develop imaging, targets, ligands, in order to image B cells in vivo in human beings and see whether you are able to deplete them in the tissues or not. And this is an area of discussion with other collaborators.
Okay, thanks. I have another personal question, and then we move to the one from the chat. So maybe we can alternate between Ziad and Anthony so that we are covering both topics. So the next question for me is again for Ziad. I like your approach of working on post-myocardial infarction and not on the classical approach of Paul Ricker working, for example, on secondary prevention. I think that targeting immune response for secondary prevention is debatable. And in fact, for example, you show that with low dose of IL-2, you are interfering with the CD28, CD86 pathway, which is extremely important, for example, from the pharmacological point of view, even in cancer, where we do the opposite. We target CTLA4 with checkpoint inhibitors. So it's risky working with these pathways because you can have, of course, immunosuppression from one end, but then enhance the risk of cancer or infection. So what is your overall view? Do you see the future of targeting immune response in cardiovascular disease more for prevention or more for acute treatment as a cure?
Yeah, you're right. It depends, it depends on how you are targeting the immune response. So in terms of this, you are right. In terms of potential side effects, whether in cancer or infections, it is important to if you are doing this in a very chronic, in a long-term manner, it is very important to be sure that you are not impacting responses to infections or responses to tumor immunity. This is why we, in fact, with the current available therapies, we were targeting very specific windows, short windows after an acute event, in order to modulate the immune response during this period and try to reestablish some balance in the immune response in a very defined period of time, within which we know that regulation of the immune response is important for subsequent development of the disease. So in this case, with rituximab, it's a single infusion. The B cells, as I showed you, the 200 milligrams will have reconstitution and recovering of their B cells during the 6-month period. And this might be enough, to regulate the immune response during this time period. With low dose interleukin-2, it's the same. We want to promote Tregs during a defined period of time. And this is why we focus on this few months after acute coronary syndromes. What is interesting with the Tregs is that, contrary to canakinumab, for example, to the IL-1 beta pathway, we know that modulating Tregs has no potential great impact on responses to infections, because you leave, if you have an infection and you have an overactivation of the immune system, it will overcome your Treg activation by the low-dose interleukin-2, which is not the case when you have hugely blocking antibodies against major cytokines like IL-1 beta. Now, if you want to go to long-term treatment of cardiovascular disease modifying immunity, I think that subtle modification of the immune system, like tolerization or vaccination, would be much more appropriate than a biologic that would substantially shut down immune responses. Okay,
thank you. So maybe we can move to some questions from the chat. Anthony, there is one question from Hamed asking if there are any drugs targeting apelin that have been approved.
No, not yet. So there is still no clinically approved compounds for apelin agonists or antagonists. There's probably one or two which are in clinical development.
Okay, perfect. And we have another question about actually the expression of the molecule. Is this just located on vessel surface, endothelial cells, or is expressed anywhere else?
So GPCRs in general will be synthesized intracellularly, but they will have to be on the cell surface in order to bind water-soluble ligands. Generally, the way in which you can check that they are on the cell surfaces, as we've shown, is to use a cell surface marker, although you can permeabilize cells to get some idea of what the relative distribution is of the receptors. It can be that these variants, these single amino acid variants, if they occur in an area where there is a post-translational modification, where there's glycosylation of a particular amino acid. These are critical for stabilizing a G protein coupled receptor in the cell membrane. So this can mean if you have a variant of a different amino acid, then they may not make it to the cell surface. And the consequence of that is the same if the variant doesn't get to the cell surface. You won't see any pharmacological or physiological effect.
Okay, thanks. And there is one question from myself. You have shown that MM07 is clearly affecting remodeling of small vessels. So have you done any work on myocardial infarction on top of pulmonary hypertension?
No, we've just stayed with pulmonary arterial hypertension, partly because of the pathway into the clinic. One of the advantages of studying a rare disease is that there is an accelerated pathway through the Federal Drugs Administration, and therefore you can get status for approval much more easily than looking at something in terms of the heart. And secondly, in the UK, there is a small number of centres to which the patients come to. And that means that if you're carrying out a clinical trial, these can be done very rapidly, and you can access the relevant patient population by engaging with those who run those clinical trials, so that you can get all of those patients quite quickly from the different centres. So these are the two reasons for really focusing on pulmonary arterial hypertension, although I agree it's likely that apelin agonists, given its very powerful inotropic and beneficial effects on the heart, are likely to be effective in other cardiovascular conditions.
Okay, excellent, thanks. Ziad, we have another couple of questions from Hamed from the chat for you. And one is asking if rituximab has been approved for cardiovascular disorder, but probably the answer is no.
Well, it's as I said, it's contraindicated.
Yeah.
So until we show that it has beneficial effects in preventing clinical outcomes, it will not be approved.
Perfect. And there are another two similar questions about the concomitant treatment in your trial. So they are asking why they are given paracetamol and corticosteroids, and also if the patients were treated with anticoagulant drugs.
Yeah, so for the first question, I mean, the use of methylprednisolone is because this is what is recommended with every first injection of rituximab to prevent any potential allergic reactions. So all the groups, even the placebo group, will be receiving methylprednisolone. So the only difference between the groups will be the injection of rituximab. We don't know of any either beneficial or detrimental impact of 100 milligram of methylprednisolone in patients with acute coronary syndrome. So this was allowed by, in terms of scientific rationale and ethical rationale, there is no potential harm from methylprednisolone injection. The other things is that, yeah, I mean, all the patients receive all the approved treatments for acute myocardial infarction. So of course they have the appropriate anticoagulants, they have the appropriate other medications that are indicated for their treatment.
And yeah, there is one last question from the chat asking where plasma B cells go after rituximab.
They go nowhere. They are still there, and they are not depleted by rituximab. So all plasma cells that you have developed before the injection of rituximab will stay there for life because they are long-lived. However, during the period that B cells are depleted, so this 6-month period, a new generation of plasma cells will be hampered, will be prevented, and new generation of antibodies will be prevented.
Okay, excellent. We have some questions from the panel. Carlo, please. You can unmute yourself and ask your question.
Ziad, I really enjoyed your presentation. However, you, as many others, quote the canakinumab trial and the colchicine trial as providing solid evidence that inflammation is important in cardiovascular disease and that anti-inflammatory drugs do work in preventing atherothrombotic complications. Well, if you look carefully at these trials, in the canakinumab trial, CANTOS, you get an effect of canakinumab, which is of borderline statistical significance with the intermediate dose. But if you look at the higher dose, double that, then you lose statistical significance. So you don't clearly see dose dependence of the protective effect. And as I say, the main effect is statistically uncertain. Then if you look at the colchicine trials, in the trial where CRP levels were measured, there was no change in CRP levels. So what is the evidence that colchicine, whatever it does in that setting, acts by reducing coronary inflammation?
Yeah, so I agree with you, Carlo, with colchicine and we should remind ourselves also that colchicine had no impact on hard outcomes like mortality, etc. It had even a small trend towards the opposite direction. But colchicine has also many other impact than just on the NLRP3 inflammasome pathway, and may have modulated the disease process through other ways. In terms of canakinumab, I agree with what you said. However, if you look at, we know that canakinumab has worked if CRP has been reduced, right? Because this is the IL-1, IL-6 pathways. And the authors have shown that if you look at the biological efficacy of canakinumab, so the patients who had reduction of CRP, they have benefited from the drug in terms of reduction of cardiovascular events. So in terms of biological actions and relation between the biological activity and cardiovascular events, there is a clear relationship. The patients who have reduction of CRP under canakinumab have reduction in their cardiovascular events. So
I'm familiar with
this
analysis, but this is post-hoc subgroup analysis, which has
I know. I'm trying
limitations.
I'm trying.
Actually, Ziad, Carlo, in any of his talk, when he discussed about canakinumab, he says you can torture the data as long as
but I would like.
They will confess. If you torture the data, they will confess.
I would like to take the opportunity here to say that the inflammation in cardiovascular diseases is not only related to IL-1 and IL-6.
Sure.
We all.
That's why I like your approach.
We all the time talk about CRP levels, okay? And this is very important. We forget that, and we tend to say that if you don't have elevation of high sensitivity CRP, then you don't have residual inflammation. This is totally wrong. CRP is very, very specific to IL-6. So, I need to repeat this again, and I may need to write it somewhere, is that, when you have human beings with selective mutations in the IL-6 pathways, have zero CRP in their blood, despite the fact that they have still other pathways of inflammation, they have zero CRP when they have single mutations in the IL-6 pathway. So CRP is extremely specific for the IL-6 pathway. It doesn't mean that if you don't have other type of inflammation, you may have other type of inflammation that are completely independent of IL-6 and CRP. So this is important to remind.
But actually, Carlo, these are important points that you're raising. The reality is that immune response is extremely complex, and we are still working in a sort of very conservative manner, using the same monoclonal antibody in different pathologies, where we should know better about the mechanism and target, for example, the vascular immune interplay in a more selective and specific manner. So there is still a lot of work to do, but clearly it's the horizon is promising. We have Amrita with some questions.
Hi, thank you. I thought the talks were fantastic. I've got a few questions for both of you, actually. So first of all, Ziad, the rituximab specifically is contraindicated in STEMI patients for what? Or is it a general because of an allergic? You mentioned some
we really don't know why it is contraindicated. It is because, you know, when this was approved for use in cancer and in autoimmune mediated diseases, this was a contraindication. We don't know why, but, you know, since there is this contraindication, you cannot go and use it as you want, and then you have to go through very important, you know, and careful safety trials, etc., and then in order to be able to define a dose, etc. But we really don't know.
Yeah, and it's
but for the IL-2, the high doses of IL-2 were associated with what we call capillary leak syndrome, with very high doses of IL-2, and this is a very good scientific reason not to use high doses of IL-2 in patients with acute poisoning.
But it sort of triggered in me a thought that perhaps the guide, in terms of potential side effects and understanding the mechanisms of those side effects, is something that could be better developed, at least if in a cardiovascular focused database. The immune therapy in cardiovascular disease, the interest in it, of course, has exploded. And actually understanding specifically what targeting, whether you're giving ligands or whether you're blocking the receptors and the targets, what impact that has for specifically cells of the cardiovascular system is information that's missing. I thought it was really insightful, Ziad, that you went to the pages and actually you couldn't see the information that was relevant for cardiovascular disease. And so this is an area which clearly we need to be adding to when it comes to the guide. And it's an opportunity, of course, that comes from developing a cardiovascular specialized portal into the database, because it requires the bringing together of individuals who have that information, and then of course the adding of that information to the guide. I am sure that that's what happened with the immunopharmacology database. And actually I was going to ask Steve and Jamie, so the creation of that portal must have led to some of the expansion of the information and data that Ziad highlighted. And that has to have come, I would have imagined, from developing the immunopharmacology portal. Is that correct?
Yes, it is correct. It came from developing the portal, and it also came, indirectly, but it came from engaging with the community. I think that was the engagement with the immunological community was actually really important in the way that we were adding things to the guide, and adding things that are of interest to them that we'd have missed out before. Again, particularly in clinical directions that were to do with inflammation, for example.
And I think actually an area
one of the things that did come out of that was not just the focus on targets and ligands, but the expansion into cells and processes.
Yeah.
Clearly, that's a very important part of how immunopharmacology works. It's not just about those interactions, it's the cellular interactions as well. And that was one of the expansions that was adopted as the discussion with immunopharmacologists continued.
And of course, one of the key difficulties with cardiovascular disease is that it's chronic. And so when we're targeting the immune system specifically, or indeed when we're giving drugs that mimic apelin, then you have to be considering the long-term effects of those treatments and how they might impact on other pathways, immune responses, on blood pressure. Anthony, I was thinking, you know, the apelin drugs and their prominent effect on vascular reactivity outside of the relevant vascular beds for pulmonary hypertension. Is that potentially a limiting factor of the benefit of those drugs in the setting of PAH?
I don't think so. I think apelin is a Goldilocks drug in the sense that it does a certain amount of vasodilatation, but not too much. If you were to compare apelin against, say, inhibiting nitric oxide, then, or you manipulated the nitric oxide system, then you would see much greater vasodilatory responses. So I don't think that it would have long-term effects in the way that you might be suggesting. We have a parallel program funded by Cancer Research UK, which is to look at antagonists in glioblastoma. And in that case, we're blocking the apelin receptor. And in those studies have been going on where you actually grow the human glioblastoma in the animal for a period of time. And this is up to about 7 or 8 months. And you don't see any detrimental effect on these animals by blocking the apelin pathway. So I don't think it's going to be sort of maybe some initial effects on blood pressure, but I don't think there will be long term, because I don't think it's such a dramatic vasodilator as others might be.
And the variants you found, they're rare variants, right? So that is less than 1%. So is this something that would be, are you thinking, I mean, PAH is, I suppose, rare already. So would this be a precision medicine approach that you would take into your PAH patients? Is that what you're potentially advocating?
I think the precision medicine approach would be for bone morphometric receptor type 2, simply because we know that those patients generally have very much reduced apelin and elabella. So that's how those patients would be recruited, and probably simply on the basis of that variant. And they're being genotyped anyway, so you get round some of the potential problems with precision medicine studies. The reason for looking at the variants was really to try and identify individuals where we can't do knockout studies in humans, but we can let nature do this for us. So accidents of nature are really good to identify and then explore in terms of pharmacology. And as I said, the key point about the 100,000 genomes is that, unlike competitor databases, they're anonymized and you can't recall the patients. We can recall the patients, and the advantage there is that, for example, the apelin receptor is present on platelets because it has antithrombotic effects, so that we would be able to look at apelin binding in those particular individuals who we've identified. We were poised to do this, and then the pandemic hit, and then everything stopped. And this is one of the problems that we have, that we've made good progress in actually identifying them. But I think that's the exciting thing, that you may be able to also recall the patient and carry out forearm blood flow studies as well. So this is why I think that identifying groups in that particular cohort is a way in which you can make progress in trying to get a genetic signal. And as many people are aware now, industry likes to have, or really almost requires, some form of genetic information in addition to some pilot studies in humans before they will consider a particular drug target. And this is one way that one might be able to identify particular pathways that may be important.
Anthony, given that we are with you, before moving to Tom Gudzik, who has the hands on, there is another question from the chat saying that apelin is expressed in neurons and the adipose tissue. Do you expect any side effect?
No, because apelin does not cross the blood-brain barrier. So there is a very large, as the person has said, distribution of apelin, but probably more elabella in the brain, and there are apelin receptors present on neuronal cells. In the periphery, there's not really very much that we can detect. In terms of adipose tissue, we've actually looked at very long-term infusions into patients with type 2 diabetes, but we haven't really seen a great beneficial effect of very high concentrations of apelin or different concentrations of apelin on that. So we're not certain that we would actually necessarily see any signal, because we have measured a lot of biochemical parameters in those individuals and not seen any changes.
Okay, fantastic. Tom.
So I raised my hand before the previous discussion on the firstly complexity of the immune and inflammatory response in cardiovascular disease and the potential selection for best targets. And I think it's a question in some way to all of the speakers, or four. As with such a complex systems that we or regulations that we experience in cardiovascular disease, what is the process for choosing the target? And it's maybe we can start the question with Ziad. Do you base this on mainly the combination of human and animal experiments to decide to use certain medication rather than the other? Because I think one of the problems is what we target in the future, and whether we can define a new target, and whether our guide to cardiovascular pharmacology, if created, what should it contain to help in such decisions in the future, in selecting the targets. The example of 100,000 human genome usage for fine-tuning the target is very good. UK Biobank provides quite a lot of data allowing us to verify the validity of certain pathways and targets, and this is exponentially growing. But I wonder, from your experience of actually doing these trials and making these decisions, how does this decision come about? Why do you choose IL-2 or rituximab, for example?
Yeah, this is a very important question. I don't think that information from GWAS would be relevant to acute responses to an acute event, right? The GWAS information is very important in chronic disease setting, right? I don't think that acute immune responses can be reflected from GWAS studies. I mean, we know that, for example, responses to vaccination in identical twins are very different, right? So you need to separate this from the chronic disease when you are targeting the acute disease setting. The other thing is that, of course, what we want to see is that when we have data, preclinical data, that reproducible data in human settings are there too. For example, when I'm talking about B cells and BAF signaling, we showed in the same paper that by analyzing a registry of patients with ST elevation MI, that BAF levels in the blood were related to cardiovascular events in these patients in the 6 months. So this is how you try to add supportive evidence, if it exists, in order to move forward to clinical translation. And when you have this information and you know that this drug exists, so why don't you try it and see how it really impacts the immune system in human beings in that condition? So you have to accumulate in these acute settings data from preclinical data, but also early translational data in human beings. And if you can have additional GWAS data, of course it would be interesting. But again, I'm pretty, I'm very reluctant about how good are the GWAS data to acute immune reactions in some disease settings.
I mean, probably, I would argue that they would be able to predict, at least to some extent, the sort of maximal ability of the organism to respond in certain ways. So I still think that with appropriate endpoints and analysis, one can utilize this also in response to acute injuries. Of course, infections or external pathogens, when they enter into equation, is a slightly different story. But I agree with you. I think it's a very good and common. But what should we do in the Guide for Cardiovascular Pharmacology that would actually help in this process? I think this is the main question, and what is feasible.
Well, in the area of immunocardiovascular interactions, I think what I mentioned is that, you know, it is very important to have curated data about, you know, ligand expression in the immune ligand expression in the cardiovascular system, immune receptor expressions in the cardiovascular system. All of this is important and is not there, you know, in the Guide to Pharmacology. So we talked about cell-cell interactions. There are a lot of cardiovascular immune cell interactions that are known and published and are being, you know, studies more and more, and this information would also be extremely helpful in such a guide. So,
if I can just jump in, we don't at the moment do very much expression, very much data on which cells express things, apart from in the immune cells themselves. But the team that look after this have got previous experience in gene expression databases. And if this was something that were very useful for the cardiovascular community, we could certainly consider bringing target expression to being part of the annotation of our targets as standard. So it's definitely doable, and it's doable by the team.
Okay, fantastic. I think we can close on this point, which is a very important point, and we will debate afterwards at the end of the meeting today. So I wish to thank all the speakers for this morning session, all of you for attending. We will stop now for 50 minutes and we will reconvene at 1 p.m. with the next session. So see you soon.
Hello, everybody. Welcome back. I hope you enjoyed your lunch break. We had a fantastic morning with some excellent talks, and I am sure that the afternoon is going to continue in the same vein. Just to remind everybody that we will endeavour to take questions after each individual talk for the remainder of the webinar. Any unanswered questions will be answered by the speakers after the meeting, and we will send those to you. So please post your questions in the chat. So without further ado, I will introduce the next speaker. It's Professor Mark Kearney. Mark is there, I think, yes. Professor Mark Kearney is Executive Dean of the Faculty of Medicine and Health at the University of Leeds, and he's also BHF Professor of Cardiovascular and Diabetes Research. And this is in addition to his busy life as a cardiologist. His work, and the work that we know him best for, of course, is his delineation of the signaling pathways of insulin and IGF-1, and his linking of those signaling pathways and mechanisms with cardiovascular disease, and how diabetes and cardiovascular disease interact. So it's with great pleasure I'll hand over to Mark. Mark, I understand that the Be There Global are going to be managing your slides, and all they'll need from you is to say next slide, please. Are you there?
In a very Chris Whitty fashion, Amrita.
Is that okay?
Yeah, that's fantastic, and a big thanks for inviting me to speak at this webinar. It looks fantastic, and thanks for that introduction. I was going to do it myself, but you've done it for me. But yeah, I am a clinician who's very interested in the link between diabetes and heart disease, and I've thought very carefully about this talk. And I think the initiative that this webinar is underpinning, I think, is fantastic. The ability to share resources, share targets, and share data to really improve the lives of our patients ultimately is fantastic. And as Amrita said, I'm very interested in using preclinical models of disease to inform new mechanisms of disease in humans and inform new therapeutic targets. So the next slide, please. So yeah, so I'm just going to briefly talk about three things in the talk today. I think there's no doubt that when I first started as a doctor a few decades ago, cardiovascular disease was very different. It was usually a one disease process. So people would have ischemic heart disease, people would have diabetes. Very few people would have diabetes when I started in medicine. But now it's a multi-morbid condition, and it's very much changing the way we need to think about disease management, disease targets, and the future. I think we've made massive strides in cardiovascular disease in managing disorders of the cardiovascular system, the heart and heart failure, which is my interest. But we have major challenges coming down the track in terms of multimorbidity. I'll talk a little bit about that. Amrita's talked to you about how I use gene-modified mice to inform new targets. So the bulk of the talk will be talking about how serendipity in science can inform new targets, and how you can move from a transgenic model into testing things in humans and human models. And I'll talk a little bit about some translational opportunities, about harnessing the power of endogenous bioactive molecules. And I'm very interested in the endothelial cell, and as you'll see as the talk goes on. So three main bits of the talk that hopefully will stimulate some interest and some discussion. So next slide, please. So really, diabetes by numbers, and every day these figures go out of date, really, because the number of patients or people across the world with diabetes is growing exponentially. And I could show you one of those pictures of the globe going red, red, red across the globe in China, in the US, and in Europe, about the number of people developing diabetes. But at the moment, if there are 400 million people with diabetes across the globe, that'll increase to 600 million in about 10 years' time. Locally in the UK, there are 4 million people with diabetes, and I think it's well accepted now that diabetes is a cardiovascular disease. It affects the heart, it affects the blood vessels, so it really is a cardiovascular disease. There are probably 4 million people in the UK who have diabetes or pre-diabetes, so they're living with the disorder without knowing. And what's driving this pandemic of disease across the globe is obesity. And I'm actually at a meeting at the Royal College of Physicians these 2 days, and I was listening to a really good talk about nutrition and how over-nutrition really is driving this pandemic of obesity and cardiovascular disease. So if you do develop type 2 diabetes, and I'm really focusing on type 2 diabetes today, you have three times increased risk of developing ischemic heart disease, so atherosclerosis and its outcomes. You probably have 3 times risk of developing heart failure. So, and I'm really talking mainly here about systolic heart failure, so a reduction in ejection fraction. Although we do have a major problem in patients coming to us now with preserved ejection fraction heart failure, where new targets and sharing data are really important, because I don't think in terms of heart failure treatment, we can wait 30 years as we have for heart failure with reduced ejection fraction to get to a point where we understand the disease and we're delivering really good treatments. And if patients do develop heart failure and they have diabetes, there's a 3 times increased risk of death despite current therapies. So the problem is here, it's stark, and it's really, really important. Next slide, please. So disease is really changing, and this is data. There's a beautiful article on the left as you look on the screen. This is data from the Lancet, a paper from John McMurray and John Cleland that looked at about 4 million people with heart failure. And this was heart failure with preserved and reduced ejection fraction. And you'll see over time, as you look on the screen, over time on the x-axis, in years, the number of comorbidities patients are suffering from has increased massively. So most patients with heart failure will have 2 or 3 comorbidities, but it can go up to 10 comorbidities. And when I talk about comorbidities, the predominant ones will be kidney dysfunction, type 2 diabetes, chronic obstructive airways disease, hypertension, but there's a huge list of these things. And that's very much changing the way that these diseases behave, so where heart disease behaves. So it's influenced by inflammation, hypertension, all of these comorbidities can impact on the behavior or the mechanisms of disease progression. And the importance here is quite stark. On the right side, as you see, it's just a paper that we looked at. We published this a few years ago. They looked at about 2,000 patients with heart failure. And it just looks on as the number of comorbidities. And it just went up to 3. And if you have 3 or more comorbidities and you have reduced ejection fraction heart failure, and this is outpatients, so this is patients with ejection fraction less than 50% who attend my clinic and go home on therapies. And if you have heart failure, reduced ejection fraction, and 3 or more comorbidities, your years of life lost over the next 10 years can be well in excess of 4 years of life, despite really good therapies if you're a man, slightly less if you're a woman. But if you look at the patients without comorbidities, if you're a woman without comorbidities and you have heart failure and you're well treated, your years of life lost are no different to what you would expect in a patient without heart failure. So comorbidities are really, really important. And this is where new data and sharing targets, I think, is really important. So next slide, please. So we just looked at some data from the UK Biobank, which is a huge resource. And it's a fantastic resource and shows the power of collaboration and sharing data. And we looked at about 4,500 volunteers. So they're not patients, but these were the subjects recruited probably about 10 years ago. And you'll see that probably it's almost fair to say that obesity or being overweight is the new normal in the UK. So 34% of patients or people were normal weight, but 43% were overweight or 23% were obese. So this is something that's really not going away, it's here to stay. And the presence of obesity and diabetes in this analysis, which is currently under revision at a journal at the moment, led to an amplification of systemic inflammation, dyslipidemia, hypertension, sympathetic activation, liver disease, and arterial disease. So these conditions that are driving this pandemic of diabetes and multimorbidity are additive. So that's really, really important. So next slide, please. So this just basically sets the scene as to where we are in terms of diabetes and multimorbidity. So it's really important. The more comorbidities you have, the more important and the prognosis is. So now we'll talk to you a little bit about some work that we've been doing in Leeds that we published a year or two ago, using the power of transgenic mice. And I think this is where transgenic mice should be used properly. It's to inform human disease and not just as a so what type of publication. So we've used transgenic mice to try and develop new ideas on how to treat our patients. So as Amrita said, that we're very interested in the insulin IGF-1 signaling pathway in the endothelium. So you see in the cartoon there that this is a model of increased IGF-1 receptor expression. So I'll show you the data in a second, but we overexpress the IGF-1 receptor specifically in the endothelium of mice to see the impact of this perturbation on the responses to different models of human disease. And I'll talk to you today about obesity. And it was a very surprising finding that led us down an avenue that really does guide us into new ways of thinking about drug targets. So we found that the endothelium of mice that overexpressed the IGF-1 receptor in the presence of obesity secreted a small molecule, and I'll show you the data in detail, that actually impacted on the gut wall. And this is where sort of whole body physiology is really important, all systems approach to science. And we found that the molecule changed the gut wall phenotype and actually impacted on the microbiota and led to possibly a change in glucose homeostasis. So next slide, please. So this mouse we published a few years ago that overexpressed the IGF-1 receptor, specifically in the endothelium, under control of the Tie2 promoter enhancer. And in our original publication, we did rigorous assessment of off-target expression that was minimal. So we treated these mice with an obesogenic diet. So it's a very short-term perturbation. So this in mice is 8 weeks perturbation, unlike you would see in a human, which is years of overnutrition. But what we found is the mouse that overexpressed the IGF-1 receptor on a standard diet, and this is the where it says lower weight gain on the graph, as you'll see, actually didn't have a difference to the wild type. So the transgenic had a similar weight on a standard chow diet. But when fed a high-fat diet, an obesogenic diet, the mouse with the endothelial cell-specific perturbation failed to put on weight, so had lower weight gain, which was really interesting. So then we assessed adipose tissue depots using MRI, and you see that both subcutaneous and visceral adipose depots in the transgenic mouse with an endothelial cell specific perturbation actually gained less weight. So this is nice data to show that it was in both subcutaneous and visceral white adipose tissue. So an endothelial cell perturbation led to a protection against weight gain in these mice. So next slide, please. So we assessed the mice in a bit more detail and to try and work out how endothelial cells might communicate with the gut or might impact on weight gain on an obesogenic diet. So we assessed fasting glucose in the mice and found that the mice with the endothelial cell perturbation had a lower glucose at rest or when fasting. And then we assessed insulin resistance using HOMA-IR, which is a product of insulin and glucose, and it's a marker of whole body insulin sensitivity. And we found that the mouse with the endothelial cell perturbation was more insulin sensitive. And then we performed a glucose tolerance test, which is injecting glucose into the mouse and following the perturbation or the increasing glucose over time. And you see that the wild type mouse went up to about 15 millimoles of glucose after the bolus, but the transgenic mouse had less expression, so a low increase in glucose in response to the glucose bolus. So good evidence that the mouse is protected against the development of obesity-induced glucose intolerance and insulin resistance. So we carried out lots of experiments to work out what the mechanism was. It was no difference in terms of metabolic rate, no difference in food ingestion. But what we did find was that the mice had a change in their gut function. So we gave the mouse a bolus of lipid and followed lipid absorption over time. So this is the graphs on the right, as you see. You can see the time post-lipid, the increase in lipid in the mouse with the endothelial perturbation was less, suggesting that they were fat absorption was reduced in this mouse. So it gives the idea that the endothelial cell was communicating somehow with the enterocyte to change fat absorption. Next slide, please. And on the left, you'll see a cartoon showing how close blood vessels are to enterocytes in gut wall lacteals. And we actually thought that it may be possible that the enterocytes were impacting on the microbiome. So we sent the mice feces to UCL Davis for a blinded assessment, and we found that the intestinal microbiota was significantly altered in the transgenic mouse. And this is just some graphs at the top there to see. You'll see changes in biodiversity or diversity in the transgenic mouse. And we found quite a lot of microbes were decreased in the transgenic mouse, and some were increased. And you probably can't see it on your screen, but some of the mice, the microbes that were increased were Akkermansia, mucophilia, which is actually been shown in humans to be very important in metabolism, and transplantation of these microbes into humans has led to a protection against type 2 diabetes. So next slide, please. So it's a proof of concept that it actually was an effect of the microbiome. We treated the mice with antibiotics to eradicate the microbiome. And you'll see here on the top panels that fasting glucose was no different on a high-fat diet in the mice treated with the antibiotics. Glucose tolerance, and you remember previously we showed that glucose tolerance was protected in these mice, was also similar in the transgenic and the wild-type mouse. And just below, you'll see some graphs to show that the microbiome diversity was also not different between the two strains of mouse, the wild type and the transgenic. So proof of concept here that actually an endothelial cell perturbation can lead to a change in the microbiome. And this is the first ever assessment of that direction of communication in a whole body model. Next slide, please. So then we looked at the possibilities of what was happening in the small intestine or the gut of the mouse, and we found that we're taking samples of the intestine, that expression of some genes important in mucus production and in terms of microbial function was altered in these mice, and that's shown on the left graph on the left as you look there. So we took then we decided how would we assess that the endothelial cell was communicating with the gut wall. So we took conditioned media and exposed KCO2 cells, a cell line that we use to assess its effect on gut wall. It's a model of enterocytes, a model of gut wall cells. And we took conditioned media, boiled and unboiled, and assessed whether their effect on gene expression. And what we found is that the boiled conditioned media actually increased expression of reg3, which is really important in antimicrobial function in the gut wall. So this boiled conditioned media, telling us that it's unlikely to be a protein, it's likely to be a small molecule, increased expression of reg3 in a cell system. So we've now got to the point where we've used a transgenic model with an endothelial cell perturbation. We've shown that its conditioned media may communicate with cells to change their phenotype and ultimately may well impact on the microbiome. So next slide, please. So here we have the cartoon on the left, and this is basically the model I've described. So you'll see that overexpressing the IGF-1 receptor, which I think in this setting is acting as a nutrient sensor, overexpression of the IGF-1 receptor, specifically endothelium, in the presence of an obesogenic diet, leads to a secretion of a small molecule, which acts on enterocytes, ultimately to change the composition of the microbiota, and that ultimately impacts on cardiometabolic homeostasis. And we went on a bit further to show that this molecule was a water-soluble molecule. So we took the aqueous sample of the conditioned media and again did the experiments in the KCO2 cells and showed that this increased expression of reg3, and that's the graph on the A graph as you see on your on the screen. And we assessed for whether there was any protein in the conditioned media that had this effect, and there was no protein. So this is a small molecule having an effect. Next slide, please. So I think we're now at a point where we can take this small molecule and assess it in a model of human disease. So I want to talk you through a system or a pathway that we're developing to test this. So we've got the transgenic mouse that overexpresses the IGF-1 receptor. We've shown that the endothelial cell of this transgenic mouse secretes a small molecule that may well change gut wall function in vivo. And what we now plan to do is to perform unbiased metabolomics to identify small molecules that will alter gut function. And then in collaboration with Donald Ingber from the US, we're going to use organ on a chip. So actually going from transgenic to organ on a chip models to assess the effect of small molecules on the gut wall in a model of the human gut under flow and with duodenal cells lining the different channels. And this approach has been approved by the FDA to assess drugs in terms of liver disease. So I think going from transgenics through to high metabolomics therapies rapidly get to the point where we are in terms of heart failure now, that's taken 30 years to get to good treatments for heart failure with reduced ejection fraction. And for instance, you could get there for patients with diabetes and heart failure with preserved ejection fraction. So next slide, please. Changing. We've used gene-modified mice in the past very successfully to develop new targets. And I think now harnessing the power of endogenous bioactive molecules and new technological approaches such as organ on a chip could accelerate these approaches to drug discovery in cardiometabolic disease. Next slide. And as always, it's important to thank people for their contribution. I think that the work that I present today was performed by lots of people. And I'd just like to say thanks for everyone who's contributed. And that's it. Thank you. You stop screen. Thank you.
Thank you very much, Mark. That was fascinating. Please, all, if you have any questions, if you post them in the chat, I can pose them to Mark. So, Mark, I was really interested in the sex differences that you noted right at the beginning of your talk.
Yeah.
And the difference in the impact of comorbidities on between the men and the women. I just wondered, when the average age of the individuals that you're looking at is that pre or postmenopausal?
What
interesting.
Yeah.
And then secondly, were the comorbidities the same in terms of which, was the comorbidity that had the most impact irrespective of sex? Was that the same in both sexes, or were there differences?
I mean, the beauty of these sort of presentations, Amrita, is that you get ideas. So we didn't weight the actual different comorbidities. So that's a really, really important point. The average age of the patients coming to the clinic nowadays, for me, is over 80. So most women are postmenopausal. We have started to look at different sex differences in heart failure, and there's no doubt a difference. And one thing we published a couple of years ago was there's a significant difference in mitochondrial function. So at a very cellular level or a subcellular level, that there is a difference in sex in sexes in heart failure. But this is a massively important area, because I think we're talking about precision medicine here. But the weighting of the different comorbidities, I think I'll have to do that. Yeah, I think it's a good question.
And so then, of course, the next question related to that is, what about sex differences in IGF-1 receptor signaling or expression? How well has that been looked at?
So in our paper that we published, that I presented there, we did the mouse difference, the sex of the mice. It was different. So the female mice, the phenotype was much less pronounced. They did have a similar pattern. So we just presented male mice in the paper. But if you want to look at the female mice, it's in the supplementary data online. But there was a significant difference. But I think that the opportunity now is to take this into looking at in humans, to look at the effect of gene polymorphisms in men and women and the microbiomes. This is where the big data sets could really help us inform and answer some of these questions.
Okay, I have loads more questions, but I won't ask them because there is some coming up in the chat. There's a question here. Is there any relationship, from Venkat Kanamalapudi, I hope I pronounced that correctly. Is there any relationship between IGF-1 receptor expression, type 2 diabetes, and age?
It's a fantastic question, and just recently, so I'll answer it in three ways. So yes, polymorphisms of the IGF-1 receptor have been linked to longevity. So the IGF-1 system is very important in age, and if you have mutations of the IGF-1 receptor, it can be linked to longevity. And there's a PNAS paper of quite a few years in Ashkenazi Jews who have favorable changes in the IGF-1 receptor system. So that's good. The second thing is that there's a paper just published using big data sets that suggests that IGF-1 resistance, and that's the first paper that's been published, I think, in a large data set of humans. We published a paper in mice 10 years ago showing that IGF-1 resistance was a feature of obesity in mice, albeit. But I think that it's emerging as being important now in type 2 diabetes in humans.
And maybe this is something that we need to think about in the guide, because a lot of the targets that we look at do change with age.
Absolutely.
Maybe there's a bit of wealth of data there, and particularly for cardiovascular. When we're thinking about cardiovascular disease, the aging process is very relevant. There's a question here from Dr. Uren, who asks an important question. The guide has data on efficacy and potency, and an important question, and it's important for human data, but an important question is, what are the best models for us to be studying the question specifically for development of identifying drug targets and therapeutics in diabetes linked with cardiovascular disease? And then should we be enriching the data that's within the database with that information? Specifically, Dr. Uren has brought up zebrafish and hamsters as potential tools.
Yeah, I think that, so in my world, really, I think the best model in mice is the standard high-fat, high-calorie diet, possibly with the addition of fructose to mimic a cafeteria diet. I think that, so my way of thinking is we've got to move more into organoids, organ on a chip, to really accelerate this field. I think there's still a place for zebrafish, hamsters to inform the science, but the testing of the science, I think we've got to move quickly into doing it into complementary models like the organ on a chip that I described. Organoids, there's a beautiful Nature paper looking at vascular organoids and the changes in diabetes using iPS cells. So I think that it's a complementary approach, Amrita, that we've got animals, but we've got to really quickly get it into human models of disease.
No, I agree with you. I mean, the liver on the chip models are the ones that are the most developed. And one of the simplest, phenotyping of hepatocytes is the secretion of albumin. Yeah. And when you take hepatocytes, primary culture hepatocytes, and look at albumin secretion in a multicellular 3D system, and I think using the systems that you showed on there, particularly from certain organizations that do the 4-cell, the quad multi-cell phenotype, the albumin secretion amounts completely different. Completely different. So it's interaction between the different cell types. There's a question here from Gian Michele Massimo has asked, the molecules that are secreted by the endothelial cells, can you characterize have you been able to characterize the downstream signaling pathways for those molecules?
Not yet. Well, that's the plan. That's the plan. Yeah.
Okay, that's to come. He's not sharing today.
We haven't got them, Amrita. We haven't done them. They haven't done the unbiased metabolomics yet, so we haven't got them. But that's the next plan. And I'm really happy to share the data with everyone once it's available.
Alex Voisey has asked, were the proof of concept studies ever compared with a germ-free model? So in the absence of the microbiome. That's a really good question. Now let me just think. We didn't do that in the paper. If you look at the paper online, we did try and do quite aggressive approaches to get rid of the microbiome. The mice didn't tolerate it very well, so the data's in the paper that it's using an antibiotic approach. But it's a really important, and this is where the animal models come in. They're very different to human models, so I think we'd really like to collaborate with people investigating the microbiome, the human microbiome. Great question. Mark, there's loads more questions. I'm just going to ask one more to fit it in, but I have promised that we will get these questions to you and get some answers and share it with the everyone who's registered after us. But this last question from Valerie Shinnikov has asked, is there an impact of changes in expression of IGF-1 receptor on the endothelial function, in particular on the formation of NO and other major vasoprotective factors? So basically, what's the mechanism of the endothelial dependency?
These
molecules.
So IGF-1 is very similar to insulin. If you stimulate an endothelial cell with IGF-1, you'll get an increase in nitric oxide production. And then if you knock down the IGF-1 receptor in the endothelium, you get less nitric oxide in response to IGF-1. It's very complicated though, because, and I'm not going to go into it too much detail, but the IGF-1 receptor forms hybrid receptors with the insulin receptor, and the hybrid receptors we've got a small molecule. You can look at it. It's on Bioresource actually, that inhibits hybrid formation. So the biology of this system is much more complicated by the fact that they form these hybrids. So standard IGF-1 increases nitric oxide production in an AKT dependent fashion. You knock down the IGF-1 in an endothelial cell, it reduces that. But actually in vivo and in a good model, it's much more complicated because of these hybrids.
Look, Mark, thank you very much for a fantastic talk. I think actually in your talk this morning, there was some suggestion that a cardiovascular portal, that it would benefit cardiovascular researchers if we expanded on cell-to-cell interactions, on the distribution of expression of receptors. And clearly here you've shown that the IGF-1 receptor on the endothelial cells specifically is key. And so I think this is food for thought for thinking about how we might expand the portal going forward. I think
you're on something. I won't keep you too long, but this cell-to-cell communication, Amrita, is really, really important in any sort of resource going forward. We should be thinking about this, because this is probably what's driving a lot of the comorbidities, isn't it? The tissue communication.
Wonderful. Thank you very much, Mark. I'll move on to the next speaker. Thank you. So
thank you. Brilliant.
Next speaker is Professor Thomas Lucia, and Professor Lucia is currently Director of Research, Education and Development and Consultant Cardiologist at the Royal Brompton and Harefield Hospitals. He's also Professor of Cardiology at Imperial College and King's College, London. He also holds a part-time commitment as Chairman of the Centre for Molecular Cardiology at the University of Zurich. A man with many roles. I think we all know Professor Lucia's research. He's played a massive role in our understanding of the importance of the endothelium, what goes wrong with it in cardiovascular disease. And of course, he was at the helm of the European Heart Journal for many, many years and saw it to its current very strong, successful position. So, Professor Lucia, wonderful to have you here. I'll hand over to you now.
Well, thank you very much for this kind introduction. I'm going to give a bit of a easier talk after all this fantastic science, and move a little bit from history to the future, if you allow me to. These are my conflicts. I worked with many companies in the past. I would like to show you a little bit how we moved from herbs to mods, to biologics, and then, of course, genetic tools, and eventually from treatment, which is currently the major focus of cardiovascular medicine, to cure. Of course, you all know that withering started with herbs, and that's how cardiovascular pharmacology really started. And at the time they published books, and I wouldn't have accepted that because it probably was too long, but it made history anyway. But it took 147 years to come up with the structure of this very drug. And then, of course, the mods were the next step. And by accident, Alexander Fleming, who was not too careful with his molds, discovered after his holidays that, in fact, there was something in this that might be a cure, and others then isolated penicillin and got the Nobel Prize for it in 1928 and later. And in Switzerland, at Basel at the time, as Ciba-Geigy, now Novartis, they asked their collaborators, their employees, to bring any sort of ground from the north, and they got a fungus that the two guys that fighted among each other about priority and therefore missed the Nobel Prize discovered cyclosporine. That was a major change, a revolution in renal transplantation and eventually heart transplantation. And of course, for us specifically in everyday life, this man, Akira Endo, that would have deserved the Nobel Prize in my mind, he was inspired actually by Fleming's success with molds, and also looked at thousands, 3,800 strains of fungi, and came up with the first statins that revolutionized cardiovascular medicine. And then, of course, snakes were also a source for pharmacotherapy, as Sir John Wayne showed that ACE is actually in the pulmonary circulation with Salvador Moncada, both Nobel Prize awardees in principle, but only one of them got it. And they then inspired Cushman and Ondetti to find tetraprotide, and later any ACE inhibitor that we use today. And finally, another Englishman, Sir James Black, came up with a slight modification, learning from nature or physiology, and discovered propranolol, and eventually the whole theory of receptor pharmacology. But then we moved to biologics, and it happened primarily in rheumatology, inspired by vaccination, antibody interaction. They discovered infliximab that eventually was primarily used in rheumatoid arthritis. I don't have the time to go into this, but we have studied this actually on endothelial function. And in fact, in rheumatoid arthritis, where this is markedly decreased, infliximab can almost normalize endothelial function, suggesting that really TNF also affects the cardiovascular system and is responsible for the twofold increase in myocardial infarction in this patient group. And then, of course, the next step came with inflammation as a major driver of cardiovascular disease, with the NLRP3, a large molecule, ends up with interleukins, interleukin-1 beta, alpha, and interleukin-6, that in the liver then produces CRP. That's a readout that we use in clinical practice because it's easy to measure and rather stable. But of course, CRP is not any mediator. But nevertheless, this has inspired Paul Ritker and Peter Libby to use canakinumab, and we have discussed that this morning to interfere with interleukin-1 beta. Now, Carlo Patrono wasn't too happy with the event, with the change in event rates in this very trial, and overall it is actually 17%, as you can see. It is not borderline significant, I would say. It's 0.005, so it's quite good, and it's consistent, but it wasn't convincing enough to convince Novartis to go ahead in cardiovascular medicine. They will develop it in cancer, I hear. But one observation that we published in the European Heart Journal at that time that was very interesting is that those patients in the red curve that actually had a marked suppression of interleukin-6 had a much bigger effect size. And this is in line with what we did in the CAS lab. We did a lot of studies on cytokines at the site of coronary occlusion. And you can see here that we took an aortic sample, and at the culprit lesion, we took another sample with a special catheter, Percusurge catheter, with a distal balloon. We could actually close this segment and get the platelets, the coagulation, the serum and plasma out of it. And what we found is that interleukin-6 is hugely upregulated at the site of coronary occlusion in patients with STEMI at the time of the event. This is acutely at any time in 24 hours when patient arrived. And serum amyloid A was also markedly increased. And interestingly, hsCRP was not affected because, of course, it's produced by the liver, not locally where the action happens. It's just a readout. And we used LPA as an inflow marker, and you could see that this system really works in the CAS lab. It's fantastic to do experiments there. And then, of course, we could show in the thrombus that we removed with a suction catheter, an export catheter, that interleukin-6 is also expressed in the cells around it, the thrombus. And this, of course, needs to eventually leads to neutrophil extracellular traps of the neutrophils and a very solid clot that is typical for STEMIs. And so this really set the stage also for the next step, namely interleukin-6 inhibition, rather in 1 beta, in the rescue trial. And you can see dose dependently this antibody markedly reduces CRP, and currently a large phase 3 trial is planned. And I would be very excited if this would be positive, because it would be much in line with anything we did in the lab, in the CAS lab, and also derived from Mendelian randomization studies. The other aspect, of course, is the mutation of PCSK9. And this is a very interesting study because this paper was published in Nature Genetics in 2003. Initially, a mutation that led to autosomal dominant hypercholesteremia. But soon it was noted that there also are missense mutations, and I will come back to that. In the Glagov trial, and this is really historic in my mind, they did IVUS of the lesions in the patients treated with a PCSK9 inhibitor. And what they showed is that, in fact, here the line of identity above it is progression, below it with the minus signs its regression. And you can see that with an LDL-C of 50 milligrams per deciliter, or around 1.4, as now proposed by the ESC guidelines, for the first time, we can regress atherosclerotic plaque. A fantastic progress in this most important disease. And this led to these trials that, of course, are very short, 36 months and 4 years, respectively. But it's amazing that from 2002 to 2003 and 2018, 16 years from basic science to a phase 3 trial. Now, a major problem in cardiovascular medicine is cardiogenic shock. And we have no real remedy for it. It still has a mortality of 40%. If the patient can say hello to the operator in primary PCI setting, it's a mortality of 2 to 3%. If he can no longer speak, it's 50 to 60%, depending on the presentation. And of course, also the Impella pump so far has, in the registries at least, very negative results with increased mortality. The DANGER trial, currently ongoing, will show whether it will work at all. But this is a very interesting thing. There is an enzyme called DPP3, dipeptidable peptidase 3, and this digests angiotensin II and has negative inotropic effects. And you can see on the left-hand side in the solid lines, there is, of course, when you do a coronary ligation in a mouse, you get a marked reduction in shortening a fraction of the left ventricle. But if you then add an antibody against DPP3, you can actually restore function completely. And on the right, you have preliminary data showing that the levels of DPP3 in small series of patients actually are very, very predictive of cardiovascular events. We have now done this in 5,000 patients currently submitted, and therefore I cannot show it, but it's highly predictive, DPP3 in cardiogenic shock, and maybe that's a new pharmacological treatment. And now come the genetic tools, and this is really a revolution in pharmacotherapy. It started, of course, with a decade of genome-wide association studies for coronary artery disease and for lipids and all many other diseases that are relevant in cardiovascular medicine. And with that as a background, these could be, of course, therapeutic targets. But the major breakthrough came by this two gentlemen, Andrew Fire and Craig Mello, who got the Nobel Prize in Physiology in 2006. And for their Cell paper on the right, they showed for the first time that in C. elegans, they could actually silence genes, transcription from the transcript to the protein using double-stranded RNAs called RNA interference. And how does this work? Well, the revolution in pharmacotherapy has two pillars, the antisense oligonucleotides and the siRNA that we have used in the lab in our mice for more than a decade, but now it is ready for prime time. Antisense, of course, technology is single-stranded. It blocks the transcription at the site of RNAs, as shown here, while the double-stranded RNA interference binds to the RISC, the RNA-inducing silencing complex, and sits there for prolonged periods of time. And this is quite the fantastic aspect of it. So it can actually inhibit the translation from the transcript to the protein for several weeks to months. This is from a review we wrote with Ulf Lampmesser, my former associate, and what works really nicely in the liver currently, which is the main target, is that it has a specific receptor called the azioglycoprotein receptor that's only expressed on liver hepatocytes at their surface. And so with GalNAc, the galactoglutamine, when you link this to the double-stranded RNA, as shown here, it will then specifically bind to these hepatocytes and only inhibit the transcript in these very cells where, for instance, a PCSK9 or any other molecule of interest is produced. And then, of course, with the risk attachment, you can inhibit this in a prolonged fashion. On the other hand, the intracellular release of ASO from the endosome will then do the same thing. And of course, the first clinical paper, that's not an outcome paper, but a lipid paper, is inclisiran in patients with high cardiovascular risk with elevated LDL cholesterol by Karl Schrey and Ulf Lampmesser. And they show basically on the left that indeed, amazingly, for almost 6 months, there's a significant reduction in PCSK9 plasma levels, as well as a change in LDL cholesterol of similar magnitude, which is quite substantial. This is on top of statins, obviously. And of course, statins increase the number of LDL receptors, and then these drugs in general work much better than if you give it alone. But I think that this is really a revolution in pharmacotherapy because it's long-acting, it's very specific, tissue-specific, and we will pursue this further. The question is, of course, is it safe? It's a big question. And here, Ulf has, with Kosh and John Castelline, has published some aspects on a possible pro-inflammatory role. As an example, TNF-alpha has been tested among many others. And you can see there is no relation to the dose of this double-stranded RNA on this particular cytokine, but on many others as well. And of course, there's a large randomized phase 3 trial with 15,000 patients on the way, and we're really eager to hear what this will show. And of course, there are other candidates for that. Lipoprotein(a) runs in families. Everybody that's listening should measure that once in your lifetime because it never changes, and then you know really what your risk is beyond the LDL cholesterol. And this, of course, is important not only for coronary disease, as you can see here, but also for aortic stenosis. And there's a linear increase in risk with LPA levels with coronary disease, such as myocardial infarction and sudden deaths, as well as aortic stenosis. With very low levels, there's a possibility of an increased risk of diabetes. In the Fourier trial, they looked at this because PCSK9 inhibitors also lower lipoprotein(a) by about 15% something. And you can see here is the LDL below 120 milligrams, the lipoprotein(a), sorry, below 120 micromoles nanomoles per liter. And you can see a reduction, but even those with high lipoprotein(a), there was even a more marked reduction. And of course, there wasn't enough power to look into this in terms of outcomes. But there is not more at the horizon. This is an antisense oligonucleotide. Currently, this is also tested as a principle in the Horizon trial. And we will hear about the results very soon. And here you can see you can achieve with this antisense oligonucleotide up to 80% reduction in lipoprotein(a). I have patients that have 1,500 huge levels, and these are particularly younger patients that have an infarction in their 40s or even earlier. And it runs in families, so family history is important. So far, no treatment. And the question will be whether if we really go low with a PCSK9 inhibitor with LDL, let's say to 1.0 or even 1.4, as in some patients has occurred, whether then LPA will still be important. We'll learn about this shortly. And then finally, besides inflammation and lipids, we have the triglycerides. And there, of course, there's also progress with antisense oligonucleotides. There's one against apolipoprotein C3, which reduces this very lipoprotein by about 75%, and thus triglycerides in the same manner, a bit shorter in action, as you can see, are these antisense oligonucleotides compared to the double-stranded RNAs that block the risk. And there's another one, the ANGPL3, but there were some issues with platelets. So I don't know whether this will be further developed, but it shows also that this lipoprotein also carries triglycerides. It's a bit less of an effect if you look at the lower panel, and it's 1 month's action primarily, maybe 2 months. So it is again shorter than the RNA interference. So why is this really so important? If you are going to a physician and you have a high cholesterol and a high blood pressure, you end up with 2 to 3 drugs that you have to take for a lifetime. 365 pills a day, sometimes 2 or 3 a day, so a huge effort. With the new antibodies against PCSK9 and other targets, you have 26 injections per year. With the sRNA, currently 2 injections a year is almost like a vaccination for COVID. But of course, the eventual route should be gene editing, because what patients expect from their doctors is cure, not just a treatment in the long run. So can we move from treatment to cure? I would put to you, yes. The traditional pharmacological therapy are really moving forward to cure. And of course, these are the ladies that got the Nobel Prize a few years ago, Emmanuelle Champartier and Jennifer Doudna, with the CRISPR-Cas9 gene scissor. And this, of course, has again the basis of genetics, the Atlas of Clinical Genetics of Human Dilated Cardiomyopathy as an example. There are numbers of genes, and we can only cure these patients eventually because it's monogenetic in nature if we know the basis and if we know how to target it. And there's an example, hypertrophic cardiomyopathy, very, very bad disease in young individuals. And of course, they have a number of genetic mutations. One of them is myosin-binding protein C. And here you see a Nature paper from the Portland group showing that if you have a mutation in red in an embryo, using CRISPR-Cas9, you can exchange this with a wild type normal gene and end up with an embryo that does not have the disease. Of course, this had to be stopped at this point for ethical reason. And of course, at the Stanford group with Joseph Wu showed the very same thing, where they cut out an exon and exchanged it as well. So this is really not just one group that could do this. And then, of course, PCSK9 is the next target, because when we look at Afro-Americans that have the missense mutation rather than a mutation that leads to familial hypercholesteremia, they have a distribution of LDL cholesterol that's shifted to the right compared to the American population. And because it's from birth, they have an 85% reduction in cardiovascular events. So why don't we get rid of PCSK9? And that's what they did in primates in this Nature paper. And you can see with this gene editing, they were able to do this primarily in the liver, a little bit in the spleen, and a little bit in the adrenal glands, which is a bit of a concern maybe. And then they also looked at LDL. And so this is one shot in a lifetime, 80% reduction in LDL cholesterol in primates so far. So what about humans? Well, there has been a first trial in humans with CRISPR-Cas9 in vivo gene editing for transthyretin amyloidosis, published in the New England Journal of Medicine. 5 patients received that with this transporter tool that they have used here. And what they did is then, of course, they looked at the TTR gene editing. Was it successful? Yes. I mean, the messenger RNA expression in red dropped significantly, dose dependently, and so did the protein production from the liver again in this very case. So this is possible also in humans. There are lots of ethical questions here we might discuss, because this is one shot in a lifetime. It can also be a shot that's not on target. And that, of course, is a big concern that we have to work on. So in summary, therefore, the discovery of the genome has now reached the clinical arena, not just in the diagnosis, but also in the treatment of patients. Furthermore, also in the epigenome modulation, that's much more difficult, but also possible. So I would say we came from far, and we will get even further, and I thank you for your attention.
Well, that was a fantastically optimistic view on the future for therapeutics of cardiovascular disease. It was wonderful. And I suppose my question to you is with all of these opportunities, I mean, clearly delineated targets, their cellular expression and their ligands, I mean, whether it be a small molecule or a herb from nature, or whether it be RNAi, I mean, we need that information in a way that allows us to identify the specificity, and even more so with the silencing, the RNAi approaches. I mean, there was a note of caution at the end, but not much throughout. I just wondered.
Well, of course, I'm an optimist, and in science we have to be optimistic. There are lots of problems ahead, I agree. So at the moment, I think it works very well in the liver, because the liver has these receptors, these azioglycoprotein receptors that are fantastic because it's quite specific. And we have to find other ligands, for instance, to the myocardium. We supported a huge grant of 30 million with the British Heart Foundation for Oxford and Harvard. They want to actually cure the cardiomyopathies, and they are using a virus that hopefully goes to the myocardium primarily. This is also an approach. It would be, of course, eventually better if we were to find a surface marker on myocytes that's only expressed there, very much like the liver. And I think this applies for any other organ, maybe the kidney, maybe in the kidney we can knock out sodium glucose transport inhibitors with a similar approach if we find specific binding sites. This is the challenge ahead, but I don't think it's unsolvable.
I can see actually Carlo's got his hand up. I can't see any questions in chat, so I'm going to let Carlo, do you want to?
Yes, Tom, very good talk as always. I know this is an academic discussion because the drug will not be further developed in the cardiovascular field. But for the record and for the audience, what you showed of CANTOS was the results on the key secondary endpoint, not the primary endpoint of the study. And if you look at the primary endpoint of the study, the upper limit of the 95% confidence interval is 0.98, which I would call and consider statistically uncertain, and I would like to see repeated. Now, there is repetition within the same trial when they use the 300 milligram dose, twice as much as 150. And with the 300 milligram dose, they failed to reach the predefined threshold for statistical significance. If this is not statistically uncertain, then we are talking different languages.
Yeah. I mean, I would now focus on interleukin-6 anyway, as I mentioned, because there the effect size was much, much bigger. And so, yeah. But, probably it was not wise to have too many dosages in an outcome trial, which of course dilutes the statistical power substantially.
Sure. Thank you.
I mean, there was a question of dose actually that came up in Ziad's presentation, and I don't know if you were present for that, and how one targets the immune system and how dose is very important, and the collation of that information, so that, I mean, when you're influencing the immune response, it is this tight balance, and it's very easy to tip it over. And And actually, my hope would be that the guide to pharmacology would be a good resource where that kind of information would be kept. I mean, when it comes to IL-6, I mean, dose, is it going to be a similar kind of issue?
Well, I mean, of course, you know, you can shut down things that are not really very important for survival, like PCSK9. You can live without it.
Yeah.
You can silence a dominant negative mutant in a cardiomyopathy because then you get actually a normal heart. But there are things that we should inhibit, but rather than block. In fact, if we inhibit too much, particularly in the immune system, we end up with more infections, and there were more infections in these trials. And this is, of course, a very, very important aspect. Of course, we can learn from the Mendelian randomization studies on, you know, how much infections do patients have that have low levels of such a cytokine. But this is much more tricky for the approach I showed.
Yeah, I think there are some really important issues about dose. Ziad, you have a question.
Yeah, thank you. Tom, great talk and overview. I would like to have your feeling and opinion about potential public acceptance of once and done gene editing for PCSK9. I mean, when we see that we have difficulties in getting acceptance for one shot of COVID-19. I mean, how in your mind can people accept once and done gene editing for life?
Yes,
disease,
you know,
where you said that, you know, with 2 injections a year, you can lower the gene, and then, you know, you don't risk lifetime potential side effects, you know?
Yeah, it's a very, very good question, and of course, this is in my mind as well. But, you know, first of all, I think we moved from herbs to nucleic acids, and this is a cultural change that is enormous, and it will go on further. And if I talk to patients and tell them that they can take tablets or 2 injections that interfere with their translation in the cell, they are all excited. I was actually amazed. I would have expected much more concerns, but maybe I see the wrong patients. And of course, there's always a part of the population that doesn't want it. You know, I have problems with statins. There are patients that are at high risk and they read all this bullshit on statins in the internet, and then they don't take it. So there is always an issue with that. But of course, with CRISPR-Cas9, we have to be really sure that we know what we're doing.
Yeah, big issues ahead. Tom, last question before we move on.
Maybe very briefly, thank you very much, Tom. Beautiful overview. But thinking about the future, I mean, is there anything we should learn from cancer, for example, where a lot of drug development has been taking place, and they have been to some extent more proactive, especially in the recent decade. And this specifically, I'm asking about maybe actually definitions of the pathology in cardiovascular medicine. I mean, HFpEF and heart failure is the prime example, where we are learning that maybe what we are identifying as pathology is not accurate, and this is actually hampering the therapy.
Oh, I mean, oncology is much further than we are. I mean, you know, I recently had a patient with mesothelioma, and I learned what sort of genes they look for to personalize the treatment is amazing. We're far from that, obviously. And we certainly have to learn more about this. But of course, you know, oncology is in a better situation. If there is a hair in the soup, in oncology, they see the soup. In cardiovascular medicine, they only see the hair, and we're not allowed to move forward. It's because we are so safe, so effective right now, that it's much more difficult than in oncology, where they accept much more side effects.
Well, okay, I think this has been a fantastic session. Lots of food for thought, and actually some interesting things to think about with regards to how we might categorize the guide for cardiovascular entry. I'm going to hand over now to Professor Tom Gucic, who's the chair of the next session. Thank you very much, Professor Lucia.
Thank you very much, Amrita. Thank you to all the participants and speakers for nice presentations and discussion. And in the next session, we will focus on, come back to pharmacology and look at two aspects: targeting guanylyl cyclases with quite a large heterogeneity in this field, and Professor Adrian Hobbs from Queen Mary University of London, whom we all know very well, will talk to us about this. And then we will hear about platelets from Steve Watson. Please, Adrian.
Thank you, Tom. Good afternoon, everyone. Okay, hopefully everybody can see my slides. Good. I'd like to try and do two things this afternoon. First of all, talk a little bit about the therapeutic targeting of guanylyl cyclases, which I think has been quite a success story over recent years. But also, because we're interested in nomenclature and thinking about the guide to pharmacology, I also want to give you some idea about the terminology around guanylyl cyclases, and also some of my experiences with trying to get this across to the field. And in light of the latter point, I've actually been co-chairing an IUPHAR GRAC, Guide to Receptors and Channels committee over the past 7 or 8 years now, to try and standardize the nomenclature around guanylyl cyclases. And I've chaired this with Scott Waldman from Thomas Jefferson in the US. And many of you will recognize some very prominent individuals who are part of that committee who've made significant contributions to our understanding of the different isoforms of guanylyl cyclase, particularly in terms of cardiovascular biology. So I'm going to start with an anecdote, actually, if I may. I was fortunate enough as a young postdoctoral fellow to work with the chap on the left here, Luis Ignarro at UCLA. And many of you will recognize these three gentlemen, and they're all Nobel Prize winners, as many of you will know. So Luis Ignarro and Fred Murad won the Nobel Prize in 1998 for the biology of nitric oxide, and Al Gilman in 1994 for G protein signaling. And actually, through several conversations with Luis Ignarro, I want you to picture a dinner conversation at a cyclic nucleotide conference in the sort of late 1970s. And these three gentlemen were discussing whether enzymes which generate cyclic nucleotides should be called guanylyl or guanylate, and likewise adenylyl or adenylate. And actually, there was no real agreement on the correct terminology. And the three of them sort of decided that it's much easier to say guanylate than guanylyl. It's a bit of a mouthful, adenylyl for that matter. And so, in fact, Luis Ignarro in particular has stuck with guanylate cyclase for many, many years. And actually, I'm so used to using that terminology that it's almost inevitable that I'll make a mistake during this presentation. But actually, guanylyl or adenylyl, in fact, is the correct terminology. And this is one of the first things that our IUPHAR committee set out to sort of straighten out. So this is a chemical schematic showing the conversion of guanosine triphosphate, or GTP, to cyclic GMP here, which is obviously catalyzed by guanylyl cyclases. And the process is actually driven by a divalent cation. Now, physiologically speaking, this is probably magnesium, but it will also be facilitated by manganese, for instance. And the Lewis acid effect of magnesium actually causes a positive charge to develop on the phosphorus atom of the terminal phosphate group of GTP. And this then allows a nucleophilic substitution from the 3' hydroxyl. Actually, it's probably an addition first and then an elimination reaction, which then causes the cyclization of the guanosine triphosphate and kicks out the pyrophosphate as a byproduct. Now, because the substrate for this reaction is actually the guanylyl functionality shown here, and not the guanylate functionality, which would include it's a conjugate base, so it would include a negatively charged oxygen, it is actually guanylyl and not guanylate cyclase. So having sort of sorted that initial nomenclature issue out, let me take you through these different members of the family. So the guanylyl cyclase family are formed of ostensibly soluble and membrane spanning isoforms, and they share a catalytic homology, both through themselves and also through adenylyl cyclase, which is also a membrane-spanning protein. Of course, adenylyl cyclase converting GTP to cyclic AMP, whereas guanylyl cyclases convert GTP to cyclic GMP. Now, actually, the terminology soluble guanylate cyclase, or SGC, even though it's used widely, is actually a misnomer, because it's emerging now that actually the enzyme is often translocated to the membrane, particularly through chaperone proteins like heat shock proteins, and it may well form scaffolds, for example, with protein kinase G or with nitric oxide synthase to facilitate NO signaling. So let me talk a little bit about the NO-sensitive guanylyl cyclase first, because I'm sure probably most of you will be familiar with this pathway, and perhaps this is the one which is often thought about when thinking about guanylyl cyclase signaling. So the NO-sensitive guanylyl cyclases are essentially responsible for the majority of known physiological actions of nitric oxide, and clearly it's the principal intracellular receptor for NO. Now, there are also three predominant isoforms of NO-sensitive guanylyl cyclase. There are two larger alpha subunits shown on the left here, and one smaller beta subunit. And it's the beta subunit which contains the heme moiety, which conveys the sensitivity to nitric oxide. Now, the active enzyme, or the active NO-sensitive guanylyl cyclases, are actually heterodimeric. So they have one beta subunit and one of the two alpha subunits. The alpha subunit combination with alpha 1 is termed GC1, and the alpha 2 subunit with the beta subunit GC2. Now, I probably don't need to tell you all on this call or on this webinar about the important umbrella of protection that nitric oxide and cyclic GMP signaling produces in the vasculature. This is a schematic just to go over that very briefly. And the NO produced by the endothelial isoform of nitric oxide synthase obviously activates the soluble, see now I've made the mistake already, the GC1 or 2 in the vascular smooth muscle cells to induce vasodilatation or lower proliferation. It also activates guanylyl cyclases in the endothelial cell to affect endothelial barrier function, and also probably to increase proliferation. And then extracellularly, it has effects on both platelets and leukocytes, also mediated via activation of guanylyl cyclase and production of cyclic GMP to prevent the activation of those cell types. Now, not only has there been a huge body of work looking at those important anti-atherogenic functions of NO cyclic GMP signaling in the vasculature, but we also have good evidence of the importance of this signaling pathway to cardiovascular health and disease through larger scale genomic and genetic studies. Now, this is just a plot from one of the earliest genome-wide association studies looking at associations with hypertension. I know it's very difficult to pick out individual genes here, but if I highlight it for you here, the Gucci genes, which encode for both the alpha 1 and beta 1 subunit, are associated with a hypertensive phenotype. And again, on the right-hand side, there's an increase of about 0.4 millimeters of mercury in terms of both systolic and diastolic blood pressure with people with variants in those two genes. And also more recently, genetic studies have shown that families with different pedigrees of Gucci mutations, for example, this one here, there's a number of individuals in this family that have died very suddenly and early from myocardial infarction. And again, particular mutations in the alpha 1 and beta 1 subunit cause a huge reduction in the expression of these enzymes in their tissue, and this is associated with much higher and earlier mortality from myocardial infarction. So a very clear link between cyclic GMP signaling, guanylyl cyclase activation, and cardiovascular disease, or inappropriate activation and cardiovascular disease. So that sort of begs the question, why don't we give NO therapeutically to recapitulate these protective activities? Well, of course we do. So for over 150 years now, we've given organic nitrates for chest pain and angina. And of course, the irony is that Alfred Nobel himself, that was exploring organic nitrates for use as explosives, actually had to take them for his angina several years later. Now, we also use inhaled nitric oxide in a sort of limited capacity, particularly in persistent pulmonary hypertension of the newborn. There is some evidence that it may be useful in adult respiratory distress syndromes and also perhaps COVID-19, although the evidence of this is much less compelling. And it would be remiss of me also just to mention that now is emerging evidence to suggest that actually inorganic nitrite and nitrate might be used as reservoirs, let's say, of nitric oxide, which can be given to patients in different disease states and produce therapeutic benefit. However, when we try and target nitric oxide directly in this way, in many respects through NO donors, if I can give that broad category, there are some very clear limitations. Firstly, individuals with cardiovascular disease tend to be tolerant or less sensitive to NO generation or production pharmacologically. Obviously, the delivery of inhaled nitric oxide has its own logistical problems. And there are also off-target or adverse events which are associated with targeting NO directly. And let me just explain that a little bit more. Under physiological conditions where constitutive nitric oxide synthase produces NO, this will activate GC1 or GC2, and this is a very cardioprotective phenotype, as I've shown you previously. Now, if we then think about increased concentrations of nitric oxide, perhaps produced by the inducible isoform of nitric oxide synthase, or for instance, under some conditions by the neuronal isoform of nitric oxide synthase, this tends to produce what I would call off-target activity. And what I'd mean by off-target is perhaps independent of guanylyl cyclase. Now, of course, some of these are very important for host defense purposes. So effects on mitochondrial respiration, DNA modification, for example, are key in terms of battling pathogens, viruses, bacteria. However, there is also the possibility, as the concentrations of NO go up, of causing tissue damage, host tissue damage. So the idea arose, of course, that if one could directly target guanylyl cyclases without producing nitric oxide, then you would be able to harness these cardioprotective effects of cyclic GMP signaling without having to worry too much about potential tissue damage. So this drove the development of soluble guanylyl cyclase stimulators. And if you take a note of the title of this slide, I've gone back to the sort of old nomenclature that I mentioned at the start, and I'll just come and touch on that in a moment. And these heterocyclic compounds were developed in the 2000s, primarily by Bayer, but more recently by Cyclerion, Merck, and Boehringer. And they're not dependent on nitric oxide release, but they do activate the enzyme directly, and they do synergize with endogenous nitric oxide to produce increased levels of cyclic GMP. They don't suffer from tolerance, and they actually activate the enzyme by binding to an allosteric site in close proximity to the heme group, and they actually increase the or binding of nitric oxide to the heme moiety, and therefore increase the production of cyclic GMP. And this is just an example of the large-scale clinical trials that have been conducted with these SGC stimulators over the past decade or so. So the compounds are licensed for pulmonary arterial hypertension. This is the compound riociguat. And this is some endpoint data from the patent 1 and the extension patent 2 trials, where riociguat caused a significant increase in 6-minute walk distance, so exercise capacity in PAH patients. And it also had a positive effect on N-terminal pro-BNP levels and also pulmonary vascular resistance. And in the follow-up or the extension study, patent 2, there's still a survival advantage in patients with taking riociguat, 3 or 4 years after first treatment. And actually, subsequent studies have also now allowed riociguat to be licensed for chronic thromboembolic pulmonary hypertension. And this is a different class, which is caused by microthrombi buildup in the pulmonary circulation. And this is actually the only licensed medication for CTEPH at present. More recently, a second soluble guanylyl cyclase stimulator, vericiguat, has now been licensed for heart failure with reduced ejection fraction. And this is the primary outcome data from the Victoria trial. And this is based on CV hospitalization and death. And you can see that vericiguat has about a 10% reduction in that primary outcome. And indeed, that's now been licensed for that condition. Interestingly, however, phase 2 studies with soluble guanylyl cyclase stimulators in HFpEF suggested a lack of efficacy. And actually, I don't believe that these are actually being followed up now into a larger scale phase 3 trial. Now, unfortunately, what's happened with these compounds is that we've actually now got an officially new class of approved medicine, which is really based on incorrect nomenclature. And hence, it was a pity that a lot of the work that we did with the committee wasn't out before these drugs were licensed. Now, I don't know whether we have any German speakers in the audience, but by speaking with colleagues at Bayer, apparently guanylyl is even harder to say in German, or with a German speaker, than it is to use the term soluble guanylate cyclase stimulators for this class of compound, unfortunately. So we're kind of stuck with that at the moment, even though the correct nomenclature is guanylyl cyclase rather than guanylate cyclase. What the production or development of direct-acting guanylate cyclase, guanylyl cyclase compounds has also done is uncovered some interesting physiology. So it appears that actually there are forms of guanylyl cyclase, or NO-sensitive guanylate cyclase, which are regulated by the redox environment and can change in cardiovascular disease. So the so-called SGC stimulators that I've just been talking about will target the iron 2 form of the enzyme, which is NO-sensitive. But in cardiovascular disease, where there's a more oxidative environment, the enzyme can be oxidized, or at least the iron atom in the heme group can be oxidized to iron 3. And this can actually be targeted by a different class of compound called SGC activators. And so these can activate the form of the enzyme, which is predominant in vessels in cardiovascular disease and other organs in cardiovascular disease. There is the possibility now of actually using these compounds to specifically target diseased vessels and organs, rather than a very general stimulation of NO-sensitive guanylyl cyclase. And actually, in terms of direct-acting GC stimulators and activators, I guess the world is their oyster in many respects. So one can imagine there's different organs and tissues within the cardiovascular system. These compounds may well have very important beneficial pharmacological activity. Now, clearly it's been shown in the heart and blood vessels already, but there are obvious other examples in adipose tissue, in the kidneys, and a lot of this is being driven by pharmaceutical companies developing soluble guanylate cyclase stimulators which accumulate in particular organs or tissues. So for instance, in the CNS, there's a possibility they may be very important, or could be important, in returning memory function. So certainly there's a huge scope for future development of soluble guanylate cyclase stimulators. So I just want to come back now to my original diagram and talk a little bit about these membrane spanning guanylyl cyclase enzymes, which often, I think for some reason, people seem to overlook when thinking about cyclic GMP signaling. And often the enzymes can be termed G-cyclase as a kind of coverall, but in fact G-cyclase really is used for the NO-sensitive enzymes. And actually the membrane spanning guanylyl cyclases, there's a whole family of these enzymes, or enzyme coupled receptors. So GCA and GCB on the left here are sensitive to natriuretic peptides. So HRL natriuretic peptide, AMP, brain natriuretic peptide, BNP, and C-type natriuretic peptide, CMP. But there are others. There's a largely gut-specific GCC, which is important and responsive to gut hormones, such as guanylin and uroguanylin. And there are also specific photoreceptors, which are important in visual transduction, which also produce cyclic GMP, which directly regulate cyclic GMP gated ion channels. Now, also in rodents, there are other forms of membrane spanning guanylyl cyclase D and G, which are very much involved in sensory perception, but these are pseudogenes in humans, and so the importance in human biology is still not clear. So I think many of you will be, in this audience, will be familiar with the physiology of ANP and BNP, and via activation of GCA, so the first subtype of membrane spanning guanylyl cyclase, you must be aware that AMP and BNP are largely released from the cardiac atria and ventricles respectively, and they have a number of different actions to reduce blood volume and blood pressure. So they are natriuretic, as the name suggests, and also have effects, inhibitory effects on the aldosterone or angiotensin-aldosterone axis. They have effects in the CNS to alter salt and water intake. They have direct vasodilator effects in the vasculature, and can also affect lipolysis and regulation of fat storage. And of course, again, many of you will know full well that NT-proBNP, so one of the breakdown products of BNP bioactivation, is a very well validated diagnostic and prognostic marker of cardiovascular disease. So in terms of therapeutics targeting the membrane spanning guanylyl cyclases, there was a lot of excitement in the late 90s and early 2000s when administration of recombinant BNP, nasiratide, was shown to have very positive effects in patients with acute decompensated heart failure. And BNP, nasiratide, was actually licensed by the FDA in early 2000, if I remember correctly. Now, with follow-up studies, actually this was less convincing. First of all, there was a very little effect of BNP or nasiratide on a larger scale trial, and also actually some hints that there actually may be worsening renal function with patients given nasiratide. And this is largely due to the hypertensive activity of BNP, or at least GCA activation, and effects on renal perfusion and renal function. And then indeed a retrospective analysis, a pooled analysis of randomized controlled trials with nasiratide actually came up with the idea that there's actually a risk of death, or an increased risk of death, with this treatment. So nasiratide sort of fell out of favor very quickly, as one could imagine. And I should just add at the bottom here that recombinant AMP is actually licensed for acute heart failure in Japan, albeit with some very rather unconvincing evidence as for nasiratide. But however, what does work in terms of targeting guanylyl cyclases in terms of heart failure is to, rather than give the peptides themselves as recombinants, is to actually alter the metabolism of endogenous natriuretic peptides. And this has been achieved by a drug called LCZ696, marketed as Entresto. And this is a combination of an angiotensin receptor blocker, valsartan, and an inhibitor of neutral endopeptidase, or neprilysin sacubitril. Now, NEP is important in the metabolism of all three natriuretic peptides. And obviously, inhibition of this enzyme tends to increase the endogenous levels of natriuretic peptides. And this is the primary endpoint, again, of cardiovascular death and hospitalization from the paradigm study, with LCZ against enalapril as comparator. And you can see here that there's a 20% reduction in risk with the LCZ696 over a period of 3 or 4 years. And actually the trial was terminated prematurely because of the significance of the positive activity of the drug in this patient population. So LCZ696 Entresto is now licensed for HFrEF, and actually more recently it also shows some efficacy in HFpEF, but only if you actually stratify patients, particularly those with a lower ejection fraction, and actually in females. And recently, despite the fact that it didn't meet its primary endpoint, Entresto has been licensed by the FDA for HFpEF because of this hint of efficacy in certain patient populations, or subpopulations, I should say. So I just want to come on now to the third member of the natriuretic peptide family, CNP. And this has been a particular interest of my group over the last 10 years or so. Now, CNP activates MPRB or GCB to produce cyclic GMP, so a membrane-banning cyclase, but it also activates a third member of the natriuretic peptide receptor family, MPRC. Now, MPRC is not guanylyl cyclase coupled, but actually couples to G proteins and signals through that signaling modality. So actually, this has actually raised an interesting, again, point in terms of nomenclature, because GCC and MPRC are not the same protein, and one is a guanylyl cyclase coupled receptor and one is not. And I'll just come back and touch on that in a moment. So actually, the I guess most advanced therapeutic in terms of GCB activation is actually to do with bone growth. So apologies for veering away from cardiovascular pharmacology just for a moment, but hopefully this illustrates the breadth of therapeutic opportunity in terms of targeting guanylyl cyclases. So for many years, it was known that knockout mice for GCB had a very short stature, if I can I call a mouse, it has short stature, anyway, suffers from dwarfism. And the survival of these animals is very poor, is very short-lived. And a very similar phenotype is seen in humans with mutations in GCB. So they suffer from a condition called achondroplasia, and indeed very short stature. And so again, very recently, a CNP analog, an elongated CNP, which is resistant to breakdown, has been trialed in children with achondroplasia. Again, this is just one example of how the peptide will increase bone growth in these individuals. It's not a huge effect, but it's enough to make a significant difference to their stature and I guess quality of life. And indeed, vosoritide, which is the name of the peptide, was licensed, I think earlier this year or late last year, for achondroplasia. But in addition to activating GCB, CMP, as I said, also activates MPRC. And this is some of the work that we've been conducting, I guess, or leading over the last few years. And you'd be forgiven to think that I've just put up the same slide that I showed you earlier in terms of eNOS activation and NO-sensitive guanylyl cyclase signaling, because actually CMP is released predominantly from vascular endothelial cells, and it has a number of analogous effects to nitric oxides, which will cause vasodilatation. It's anti-proliferative in vascular smooth muscle cells. It's pro-proliferative in endothelial cells. It has a positive effect on endothelial integrity, and it also dampens the activation of platelets and leukocytes. So again, it has this very broad anti-atherogenic activity in the vasculature. And in the large part, we've shown that this activation of or property or actions of CMP are dependent on activation of this GI-coupled MPRC. And indeed, a very similar story is found in the heart. So if one thinks about the production of CNP by cardiac myocytes, this will both have an autocrine effect on the cardiomyocyte to reduce hypertrophy. It has effects on cardiac fibroblasts to drop collagen synthesis and fibrosis, and also, as it does in the peripheral vasculature, has effects on coronary vasodilatation. And again, these functions are all mediated through MPRC. And indeed, this is some work that has been funded by the British Heart Foundation over the past few years. We've actually designed and developed some small molecule activators of natriuretic peptide receptor C, MPRC, that we hope will recapitulate all these beneficial activities of CMP, both in the heart and vasculature, obviously for therapeutic gain in the future. But I just wanted to come back again to kind of link the story together between guanylyl cyclase activation and nomenclature, because as I've just said, targeting MPRC, which is a GI-coupled receptor, has benefits in the cardiovascular system, but actually targeting GCC, which is the third member of the guanylyl cyclase family, so signals via cyclic GMP, has also proven beneficial in terms of pharmacotherapy. So GCC is largely present on the gut epithelium, and it's important in the regulation of salt and water, particularly chloride and water secretion into the gut. And it's actually one of the receptors for heat-stable enterotoxins, which are produced by bacteria, and often with a bacterial infection, the enterotoxins will activate GCC and cause chloride and water secretion, and this causes the diarrhea associated, for example, with an E. coli infection. So a drug, linaclotide, and other analogous compounds have actually been undertaken large-scale trials to see if you can use that pathway to actually offset the development of constipation. And indeed, when you give this drug, which activates GCC, you can see here in terms of bowel movements, there's a positive effects of those drugs in patients with constipation. So even more recently, and this is something that my co-chair of the IUPHAR nomenclature committee, Scott Walden, has been working on, it's been shown that GCC is very important in the health and homeostasis in gut epithelial cells. So it's apparent that there's a sort of conveyor belt of gut epithelial cells which constantly renews the lining of the gut. And without the activation of GCC and the involvement of GCC, there's an increased oncogenesis in these cells, which can then drive the initiation of polyps and of colorectal cancer. And in fact, Scott has been leading again the way in actually now looking at linaclotide in patients with colorectal cancer. So again, apologies, it's not a cardiovascular indication, but hopefully this is another groundbreaking new therapy that will come onto the market, which is targeted at guanylyl cyclases. So just to summarize today, hopefully I've shown you that guanylyl cyclases are a family of cyclic GMP synthesizing enzymes with huge therapeutic potential, and not just in cardiovascular disease, but above and beyond that. And I've given you examples in the gut and in bone growth. So there are both NO and natriuretic peptide sensitive enzymes, as well as others, for instance, guanylin and uroguanylin in the gut. And in fact, what we also need to think about in terms of guanylyl cyclase activations is that drug combinations targeting different guanylyl cyclase enzymes, or indeed different steps in cyclic GMP signaling. So for example, targeting guanylyl cyclase and a PDE at the same time is likely to be synergistic and perhaps give broader benefit. And we've actually produced some evidence of that in a small-scale clinical trial that we ran with a neutral endopeptidase inhibitor and a phosphodiesterase 5 inhibitor in pulmonary hypertension patients. But I think it emphasizes the utility of driving cyclic GMP production for therapeutic gain. Now, if one can avoid the systemic hypertension, which comes with too much cyclic GMP, as long as one can avoid that, you can pretty much drive this system as hard as you want, and it will therefore optimize the beneficial activity. But what I hope I've also done this afternoon is shown you that nomenclature is not straightforward, and particularly with the guanylyl cyclase field. As I said, having worked with Louis Ignarro, I often say soluble guanylate cyclase just because that's something that he's always used and said. And of course, as I've explained quite clearly, I hope GCC and MPRC aren't the same ligand, and yet I often get asked questions about the cyclic GMP signaling associated with MPRC because people assume that those enzymes are the same, one and the same. So I think the idea of having this CV-centric guide to pharmacology is really important to sort those kind of issues out. And so it obviously illustrates the need for this constant terminology, both in this basic research, for translational studies, and of course we now have a drug class based on a essentially incorrect nomenclature, and of course the production of new medicines. So I think that's me done. Thank you very much.
Thank you very much, Adrian, for a very clear overview of a very dense landscape of small molecule pharmacology, which I think is, while it's a classical approach, it is something particularly useful when we look forward. When we are talking about modern therapies, we tend to overlook that small molecule may be easiest to deliver, more stable, and have various beneficial properties that make some of the novel approaches a little bit challenging. And I encourage everyone to ask questions on the chat. We don't have any questions at the moment. We are also slightly delayed, but maybe very briefly I will ask, and if I can ask you for a brief response, that you know, the number of different molecules are licensed in relation to cyclic guanylate cyclase and in cardiovascular disease, but their uptake in clinical practice is very moderate. Where do you identify the limitations? Why these new therapies are not becoming as sort of widely accepted or even sort of tested as they could be for some of the diseases like pulmonary hypertension, where we really don't have many options?
Perhaps cost, I'm afraid, is one of the answers to that question. You know, for both heart failure and pulmonary hypertension, there are a lot of generic drugs available. So I guess sometimes the question is why move away from those cheap options which seem to work relatively well and pick up these new class of drugs. In terms of limitation, as I sort of touched on the last slide, I think, you know, pushing cyclic GMP as hard as one can pharmacologically is really a good thing. Now, you know, really the main side effect, as I mentioned, is systemic hypotension. So if one can avoid that, or at least push the pharmacotherapy to a point where that starts to kick in, you know that you've optimized the production of cyclic GMP, so to speak. And there may be further opportunity to optimize that even further by using combinations of guanylyl cyclase modifying agents, so that you may drop the dose of one, which then means that you don't affect the systemic hypertension or at least blood pressure, and then combine that with another, which then may focus the cyclic GMP production on a particular organ or vessel or vasculature. So I think there are opportunities here, and I think it's you know, again, focusing on the systemic hypotension, that's really the key side effect, which is dose limiting.
Thank you. So we don't have at the moment any further questions. As we are delayed, we will go ahead. I'm encouraging everyone to place questions on the chat, and we will forward them to Adrian and to all of our speakers. So please, if anything comes to your mind, ask. And now the second lecture in this session will be delivered by Professor Steve Watson from Birmingham, who will talk to us about targeting platelet. Steve, please, targeting platelet. Steve, please.
Thank you very much indeed for the opportunity to present today. I take it you can see my slide?
Yes.
Yeah, everything is fine. So first I want to thank Pasquale, who invited me to give this talk and for taking the time to explain the basis of the webinar. And I want to divide my talk into two sections. Firstly, I want to take a historical view of nomenclature supplements. You'll see that is somewhat self-indulgent, and to ask why they were useful in the past and why they are, how they can be useful going forward. They're certainly very useful, but we've already heard issues on nomenclature and keeping them up to date and exactly what material should go in. And I will raise that in my first half. And then I'm going to talk about platelet immune receptors as new targets for cardiovascular disease, in particular as targets that can prevent thrombosis without causing an increase in bleeding. Right, so these are my disclosures, and I think perhaps one disclosure just to mention is that both myself and Adrian are actually trustees of the British Pharmacological Society who are supporting this meeting. So I'm going to start with a picture from my first PhD student, Steve Gard. He submitted the thesis in 1989. And I don't think he knows what I did with this picture. But it's really rather a nice, simple table. My own PhD was on substance P receptors with Les Iverson, and Steve Gard was my first student and carried on working on these receptors. And it simply summarized a series of properties about the three tachykinin receptors that exist. And the reason I liked this was because Friends in Pharmacological Sciences, which was the major review journal in pharmacology at the time and still is a prominent journal, used to produce these fantastic centerfolds, and you could go almost to any lab and you would find them on fridge, freezers, notice boards. This one's actually taken from 1998. But the reason I brought these two things together is because many of the centerfolds that I saw were wrong. And also these nomenclatures, which eventually became established, at that time, there were a whole variety of other proposals of substance P receptors. This was during the time when receptors were just about to become cloned. And in fact, the best way to identify them was to look at orders of potency of agonists. So I sent this picture off to Alison Abbott at Trends in Pharmacological Sciences and said what we need is a regular update every year to address this. And this ended a year later with the first edition of the TIPS receptor nomenclature supplement. I said to Alison, why did you use a dirty yellow here? It's the gold standard. So that was the very first one. 30 pages, and I typed all of this. And in fact, I typed the first eight series editions of this. And you can see we went from 30 pages to 84 pages. Now, the way we compiled this was by writing to experts around the world, essentially how things are done today, and it's illustrated, for example, by Adrian, for the guanylyl cyclase committee. And it was quite interesting at the time because, of course, I had Bertil Fredholm and Jeff Bernstock on P1 purine receptors, the adenosine receptors, and so on and so on. And I just choose a couple of example tables from the first edition. These happen to be next to each other. One page was enough for 5-HT then. That went over to 4 pages and many more receptors. Leukotriene receptors didn't really change. But of course, the names changed, and we had this creative white space, which are publishing terms that I learned as I went along. Looking back, I've no idea how I managed to do all this typing, and I think it's a different world and how busy we are today, but I thoroughly enjoyed this. But by eight editions, I'd had enough. I think it was time to hand over, and I was now writing these with Debbie Girdlestone as opposed to Alison Abbott, and Debbie appointed John Peters and Steve Alexander to carry this on. And I now realise that Steve has been doing this for 25 years, and I hope at some stage people really recognise how much Steve has really contributed to really advancement of the field. And the whole point about this was that I didn't really mind what something was called. I just minded that we were talking about the same thing, and there were real nomenclature issues. But of course, by the 8th edition, nobody was looking for new receptors because the sequencing of the genome had happened, at least for many of the major receptors in here, and so we knew how many subtypes there were. This then followed a whole series of compendia. This was the one from RBI, Keith Watling. It's interesting that I didn't overlap with Keith, but he also trained with Les Iverson, as did Tony Harmon. Again, we didn't overlap there. We all got involved in one way or another. And Keith at times working for RBI, who eventually joined up, were taken over by Sigma. And so he produced 5 of these editions. And we've already heard from Steve Alexander, Paul Van Hoot chaired the first IUPHAR committee, 92 onwards, and the first compendia was 98, and then the next one was 2000. And then the TIPS receptor nomenclature supplement came over to the guide to receptors and ion and channels. And again, this is where Steve and John Peters were really leading on. And then this is, I think, 2015, and 475 pages, my word. I mean, it's an absolutely enormous achievement, and really many congratulations. And it's a wealth of knowledge, and I never really saw it evolving in this way, and I'm so pleased I left in 1998. But do we need a receptor compendium today, or a drug target compendium, or whatever? And of course we don't from the point of view of the receptors, because we know how many there are on the genome. That is established fact. But it's much more difficult, and particularly for some of the single transmembrane and other types of receptors, which are the endogenous ligands, and if they do have endogenous ligands, not all receptors do, how many of them? And how many are relevant? Do they have meaningful exogenous ligands? That may seem a strange question, but when you look at off-target effects of drugs, then you might think you'll understand why I put that in, and I'm going to illustrate that. What is their physiological / pathological function in mouse and human? Because it's not always the same. What are the best pharmacological reagents to study the protein of interest? And there's all this other information, genomic information, variation, protein distribution, structure, ligand binding sites. And these have become very, very educational documents, which I think are incredibly important. So, 25 years on, I've not had anybody's ever asked me anything about receptor nomenclature since 1998, until the 1st of April of this year. It was actually just after 12 o'clock, so it wasn't an April Fool's. And Steve sent me this. A weekly literature update on orphan receptors identified a paper by Ellenbass that SVEP1 is an endogenous ligand for PAIR1, a receptor that we had just published on as the receptor for heparin in platelets. Now, not everybody knows that heparin will activate platelets, but it does. And it had previously been attributed to GP2B3A, but it's actually this receptor. So Steve asked, should we include this family in the database? Well, I think it's a good question, and I'm not sure what the answer is. This is PAIR1, not very well known, 15 EGF repeat receptor. It's got a heparin binding domain. It's expressed on platelets, endothelial cells, and lung macrophages. Mediates really powerful platelet activation, but really to sulfated polysaccharides, which are obviously exogenous reagents. But what we've now shown is it mediates weak platelet activation to heparin. And in fact, it's been known for years that heparin activates platelets very weakly, and it's definitely through this receptor. But it's got it has no known role in mouse thrombosis and hemostasis models. So really, it's a receptor that we just do not know what it does. The endogenous ligand and really function is unknown. But genome-wide association studies show a strong link to cardiovascular disease, and strong may be a slight overstatement, but it's certainly a frequent gene that's picked up in relation to cardiovascular disease. So, what is SVEP1? It's the Sushi von Willebrand factor EGF and pentraxin domain containing protein. I had to find this out because, of course, I went to the database, and this is the entry for it. I think it's quite interesting. It tells me that it's a receptor it binds to 2 integrins. So it's not just a ligand for pair 1 or a receptor for these, depending on which way you want to do it, because it's a transmembrane protein. And that's the information. Now, is that helpful to me? Is that helpful? It was certainly helpful to me to know that I had other receptors. And then, is it therefore the endogenous ligand for pair 1? Well, this is taken from the paper by Ellen Bass, and SVEP1 and pair 1 causally and concordantly relate to platelet phenotypes in cardiovascular disease as determined by Mendelian randomization. Well, that is something that I'm not entirely sure of what it means, but it certainly indicates indirect evidence that they could be linked here, and that's why they went further forward. And indirect evidence is, using surface plasma and resonance, they find KDs of between 1 to 10 nanomolar. 1 nanomolar is incredibly high affinity for protein-protein interactions, particularly some of these proteins, when immobilized, effectively become almost like molecular Velcro. So this is a very high affinity and unusually high, and it definitely activates platelets. But the genotypes, the knockouts, are completely distinct. One is lethal, pair 1 has no phenotype. And indeed, I can show many examples of protein-protein interactions of uncertain significance. So is it the receptor? Should we go forward? Steve would have to ask myself and the authors of this study from Ellen Bass to fill in the box. We're not really a large committee there. We're simply going to report what is out there, but we're not really giving a critical appraisal of the data. And what are the true criteria to include this going forward? So how could a guide to cardiovascular pharmacology help platelet researchers? I've said this for many years. The British Pharmacological Society and UK researchers are focused on small molecules. You're unlikely in many cases to identify potent small molecules that block protein-protein interactions. Certainly there are good examples out there, but not necessarily for some of the proteins that we're looking at, in particularly for collagen receptor GP6, as I will show. And yet we're frequently asked to do this. There are numerous biologics that have been generated, and I was interested that Steve Alexander said there's 336 antibodies. And yet antibodies today are very much the modern day drugs. Which are the best ones? Which ligands do they block? There are antibodies to platelet immune receptors, a huge number of them. And I will show you the generation of many more related biologics. Which are the best ones? And are these the ligands of tomorrow that we should be really including? Many new types of drugs under development, particularly exciting for platelets, are protein degraders because we do not have a nucleus, but we can now degrade proteins, and they work particularly well in platelets, which have no machinery to make new proteins. Which proteins to target? Do we want to go that early on? Because, of course, it's very speculative. And, of course, platelets now have increasing number of non-hemostatic roles, so what are the side effects? And I think what will be really useful for a lot of basic scientists is information, clinical trials, sites of interactions, and pharmacokinetics and pharmacodynamics, which are so fundamental for drug development. So the discovery of platelets in hemostasis and thrombosis is where I always like to start my talks on platelets, really because I think it just makes you aware of how science evolves. Platelets would have been identified in the mid-19th century, but the person who was really credited with identifying them as through advances in microscopy, but the person who was really credited with identifying this as the third blood corpuscle, because there are many other ideas whether bits of fibrin or bacteria, was Max Schultz in Bonn in 1865. And he published this as a 44-page paper without any subheading. And to get it accepted, he created his own journal, and it was the first paper in that journal. So anybody who's having publishing problems is another way to go about this. But it was really the paper from Bizzizarro, first published in 1881 and then in expanded form in 1882, that really led to the functional identification of platelets. And you can see a simple needle stick injury causes platelet clumping, and this is figure 8 of this truly monumental paper. What always amazes me is there were 3 things in this figure that were missed for well over 100 years. Firstly, these are platelets sticking to an intact endothelium. And even when I moved to Birmingham in the early 2000s, I said to many people, as I was going to look at platelet endothelial interactions, and they said they just don't interact. Well, this is over 100 years before. Second, of course, the endothelial cells are connected. So look, there's the platelet plug at the top, and of course the platelets are binding to other cells and leukocytes in the Why would they do that if all they do is clump together? Now, what we do know is that clumping of platelets and collagen-rich atherosclerotic plaques underlies acute coronary syndromes, as illustrated by a former British Heart Foundation chair, Michael Davies, in his seminal paper in 2000. And of course, individuals who are at risk of arterial thrombosis are treated long-term with aspirin or antiplatelet drugs, aspirin of which is the best known, but of course, P2Y12 receptor antagonists. But they all increase the risk of bleeding, which although it's only in a minority of patients, it can be life-threatening, and particularly in the elderly. There's a clear need for new antiplatelets that prevent thrombosis, but preserve hemostasis. But today, the hiatus of platelet research is on their roles beyond hemostasis. And so I'm talking of arterial in the arterial systems when we use classical hemostasis. Today, a term called vascular integrity, which we've reworded as inflammatory hemostasis in a review in JCI in 2019. And this is the idea that little pinholes in the endothelial cells are then plugged by platelets in a way that doesn't require any of the classical ways of activating platelets. So the integrin 2B3A is not needed, GP1B95 is not needed, the von Willebrand factor receptor. And this, we think, it involves different receptors in different vascular beds. But this inflammatory hemostasis, we think, is very important. And while in mouse models, we've got all of these other roles, I think a lot of them are attributed to the ability of platelets to bind to subendothelial matrix proteins at small areas of exposure and really maintain a healthy endothelium. All of these, you can argue, have pathophysiological roles. And today, the big focus is on two terms, thromboinflammation and immunothrombosis. I'm not going to define either because I can't, because they're used in different ways by different people. But both terms mean more or less the interaction of platelets with coagulation, with the endothelium, and with inflammatory processes. And it's now recognized that in many cases, the inflammation can drive platelet activation, in particular, as an example that we've worked on through upregulation of podoplannin in the subendothelial wall and activation of one of the platelet immune receptors that I'm going to mention, CLEC2. So there's a lot of thinking about what platelets may do, and that's why I call these platelet immune receptors. There's two reasons. One is I think I really want to focus the attention on platelets doing more than simple classical hemostasis. And indeed, there's a very good rationale for calling them platelet immune receptors because they signal as immune receptors do. So the one that is perhaps best known in platelets is GP6, which is known historically as a collagen receptor, though we now know it also binds fibrin and fibrinogen and many other ligands, as I shall show you. It's two immunoglobulin domains, and it's linked to the S3 receptor gamma chain, which is a covalent link to homodimer, also the signaling unit of the mast cell receptor, for example. And it has an ITAM, which is a defined set of amino acids with two conserved tyrosines, which when phosphorylated by Src kinases bind two S2 domains. This becomes molecular Velcro. This now has nanomolar affinity, and this activates the signaling cascade through another kinase BTK to PLC-GATA2. Now, we identified this in the mid-1990s, but for various reasons, because I couldn't find patients who were deficient in GP6, I hypothesized that there was a similar receptor that signaled in the same way. And this led to the discovery of CLEC2, a C-type lectin domain receptor, in around the mid-2000s, with work with Katsuri Suzuki-Inui and Angel Garcia. And it's actually got nothing to do with hemostasis, as it turns out. And this is a receptor that I think truly does mediate the role of platelets in inflammatory processes. But from a signaling point of view, it was a dream. It's similar, but it's different. Here it's got a hemitam, a single YXXL, and here SIC is cross-linking two cytosolic phosphorylated tails to generate PLC gamma 2. So down here, the events are the same. It's expressed the low affinity immune receptor, Fc gamma R2A, and I'm going to talk about this in a few minutes. But known, of course, for mediating thrombosis which is associated with the AZ COVID vaccine. And again, here it's slightly different. The ITAM, a single ITAM present on the cytosolic chain. And this is pair 1, signals in a similar way, but actually through a different tandem SH2 domain protein PI3 kinase to activate platelets through the AKT pathway. So all 4 of these receptors are interesting. They mediate powerful platelet activation, but they have different The very proximal events are different. Now I'm going to focus on GP6 because it's been the focus and the hypothesis for many years, and that it could be a really fantastic new target for antiplatelet drugs. And as I said, I spent 20 years or more looking for GP6 deficient patients in the UK, within Europe. It was only in 2013 when Diego Mezzazoni published 4 patients in Chile who are homozygous for a single nucleotide variant, which is an insert that occurs prior to the transmembrane domain. And so these individuals have no GP6. Now, we've now worked on 8 of these families by various trips to Chile and identified further individuals. But so far, there's still only 11 unrelated families. And some of these families live literally thousands of miles apart and are willing to come down to Santiago to donate their blood. Now, looking at a representative population, we estimate that this has been of that over 4,000 individuals out of a population of 18 million people are homozygous for this single nucleotide variant. And it's been around for several hundred years, probably introduced at the time of the Spanish invasion because of war and because of the disease when the population was really reduced to a very low level. And of course, it stayed there because of the Atacama Desert and because of the Andes. And in the rest of South America, looking at, I think, 17,000 people, we found 2 heterozygotes. So it's a really fascinating group of people. Are they protected from cardiovascular disease? Well, all of these 12 individuals are all below 40, and most of them are still in their early teens, and actually really do not have a bleeding diathesis as they become older. But I think it'd be really interesting to look at them. But it does indicate that GP6 could be a safe target. Now, my colleague in Paris, Martine Gendreau-Peru, has worked on GP6 or collagen for even longer than I have, set up a company, Actical Biotech, using a Fab fragment of a GP6 antibody, which is now called glensocimab. And this is a clinical trial. It's a phase 1b/2a safety trial in acute ischemic stroke, in which glensocimab has been given with the best standard care to 166 patients. Now, this was simply a safety trial, so it's not powered for efficacy. But the results are very, very encouraging. There's a reduction in intracellular hemorrhage. That may seem surprising, but of course, if you block platelet activation, you're going to get less of a plug and therefore less pressure. And there's a reduction in death. And so now they've gone forward with the phase 3 trial, and indeed we're going forward with a phase 2b trial in 2a trial in Birmingham, led by Mark Thomas in acute coronary syndromes. And I think the one thing that these 2 sets of data show is that GP6 is a safe target and unlikely to cause hemostasis, at least if it's targeted, on its own. The trial on the acute coronary syndromes will have to be given in combination with aspirin and P2Y12. And then it becomes a more important question in terms of safety. So how, as a pharmacologist, do these receptors activate? And this is really, my background is that of a pharmacologist. And this is a very simple slide, but GP6 is a single transmembrane protein, binds to its ligand. I'm showing an antibody here, so this could be Fc gamma R2A. And at rest, you'll find some Fc gamma R2A is phosphorylated, some is non-phosphorylated, but there's a real battle going on between kinases, Src kinases that phosphorylate, and tyrosine phosphatases that dephosphorylate. And it's very much in favor of the tyrosine phosphatases. If you cross-link them and bring them together, you've also got these SH2 domain proteins, such as Syk, which will bind through, and Src kinases, which will bind here, and will phosphorylate here. So not only will it prevent the phosphatase from dephosphorylating, it will also phosphorylate here. And that's essentially how they signal. So they signal not through a conformational change, they signal simply through clustering. Now you can model this very easily, and this is for a divalent ligand where you're always going to get a bell-shaped dose response curve. As you get really high concentrations, you'll just get a single receptor bound to a single protein, so you won't be able to cross-link it. Here's a tetravalent version of the same thing, and again, with very high concentrations, you'll get 4 receptors. The receptors will just be bound to the ligand. I've depicted the ligand as a tetravalent ligand L4, but it will just be bound to a single receptor. So And this modeling, I should say, was done by my son, who's a mathematician, and he just did this for a 13-week project at finishing university. So the problem is that we don't see bell-shaped dose response curves to many ligands. There is some that we do, but mostly we don't. And the reason why is that this model's wrong. They aren't single transmembrane receptors. They actually exist, in the case of GP6, as a mixture of monomers and dimers. This is the crystal structure of the two Ig domains of GP6, and they can see dimerization through the D2 domain. Actually, that doesn't happen in solution. So that's a very, very low affinity, and it could just be a crystal artifact. But we were able to show dimerization using co-immunoprecipitation in different differentially tagged versions of the receptor in cell lines and by Brett. But that didn't really give any indication of stoichiometry. And it's excuse me, it's only more recently as we've gone to use many of the new advanced microscopy techniques, such as single molecule photobleaching, which I'll illustrate in a minute, and fluorescence correlation spectroscopy, which I'll indicate in a few slides' time, that GP6 is a mixture of monomers and dimers. And this is how we do it. We put in the receptor at a very, very low level, and we hit it. And if one of these spots bleaches immediately, it's a monomer, and if it takes two, it's a dimer. And actually, we know it's a mixture of monomers and dimers, and as we increase expression, we get more dimers purely by the law of mass action, because you've got more receptors, so you get more coming together in association and dissociation. So it is a mixture of monomers and dimers. Also, crystallization of where collagen is like to bind using this collagen peptide shows it binds at the Ig1 domain and actually can't even cross-link the two receptors together. This becomes very nice in this nice cartoon that was written by Alex Slater in my group with Martin Gendreau-Peru in Blood at the time that manuscript appeared. The receptor is a mixture of monomers and dimers, and a ligand like collagen or collagen-related peptide, which is what CRP stands for here, will cross-link, but it can only bind one D1 domain in the dimer. But the monomer is also equally useful because all you're doing is concentrating these YXXLs group in the inner leaflet for activation. And you don't even need the D2 domain. You can delete this off, you can still get activation, but it's reduced because we don't have that degree of dimerization. That's my prediction. What we don't have for these receptors of any ligands of defined stoichiometry. All our ligands are uncertain stoichiometry. How many binding sites are in collagen or synthetic collagen, which is a mixture of monomers, dimers, trimers, and tetramers? Convoltsin, which is snake toxin, which binds with the receptors, Fc gamma R2A, we have to cross-link a monoclonal antibody or aggregate IgG. CLEC2, it's a snake toxin. K1, it's polysulfated sugars. And yet we desperately need ligands of known valencies. We need to test models. We need to develop clinically validated assays, and we need to compare results between laboratories. We can't do any of that because none of these ligands are defined. Particularly, purified snake toxins, you have to do everything by bioassay, as we do for collagen-related peptide. So with that in mind, we've set about raising nanobodies to these receptors. Now, nanobodies, I'm sure you're familiar with now, they're increasingly important, are single-chain domain antibodies. They're much smaller than antibodies. They're not small. They're still 12 to 15 kilodalton, but that's a third the size of a fab fragment. And they're made by camelids, such as llama, and they have a similar specificity to antibodies. They can actually be produced in bacteria, so we can get 20 or 30 new ones made and express them more or less at the same time. They're less immunogenic. You don't need to humanize them, and they've got greater stability. And capricizumab has made itself into the clinic for thrombotic thrombocytopenic purpura. But we developed nanobodies to GP6. We do this with VIB, a company in Belgium. And we send them recombinant Ig domains, and then we test them for binding to immobilize GP6 and for inhibition of collagen signaling. And I've highlighted 3 here. Nanobody 2 binds very well. Nanobody 28 binds very well. Nanobody 35 is the star one here. And then we've got nanobody 53 doesn't bind at all. So I've highlighted those. Now we go to this slide, inhibition of GP6 signaling. Nanobody 2 binds well, and it blocks CRP.
Nanobody 28 binds well, but doesn't block, and nanobody 53 doesn't do anything again, so it's a nice control. And you'll see that nanobody 21 and nanobody 35, the reference one here, also block just as well as nanobody 2. So we have nanobodies here that can perhaps be used to label the receptor and also perhaps be developed as novel inhibitors. Well, here's nanobody 2, and you can see it's potently blocking the binding of collagen, and it's got sub-nanomolar affinity. And it's got a very rapid on rate and this very slow off rate in the surface plasma resonance recording. Nanobody 2 will block aggregation on human plaque and blocks it beautifully. And though I haven't got time to tell you, we could use nanobody 28 here. It would do nothing, but could be used to label the collagen fibers, which we've done and published recently. And nanobody 53 I control does nothing at all. So we've got some very interesting reagents, and we've gone on to co-crystallize. And this is why I say small molecules were never really likely to target GP6. It's got a very flat, relatively uninteresting domain. And this is where the residues that bind within the collagen-related peptide, interestingly, the nanobody 2 residues do not bind in the same positions at all, but are adjacent. And it's really blocking purely through steric hindrance. Effectively, it's a big lump. And you can see it's almost as big as the first Ig1 domain. The beauty about the nanobodies is that they come, in the case of GP6, we've got 33 different classes. They've got very different sequences in their CDR3 domain. And so we've co-crystallized nanobody 35, and it binds at exactly the same site. So it's interesting to see how we could really build up a lot of information as that reactive site. And again, they're blocking by a steric effect rather than by a competitive binding. How does glensosumab work? Well, glensosumab, if I can call it, is a bigger lump. It's a fat fragment. And here it's binding, interestingly, to the D2 domain, and in particular to this peptide sequence, because we can show that by mutating out that peptide and we lose the binding. And here we've tried to draw this in a way in which you can see CRP is really prevented from really cross-linking. But because it binds at the D2 domain, the question is, does it also prevent dimerization? So this is showing that glensosumab also inhibits binding of collagen to GP6. And now I'm using the fluorescence correlation spectroscopy method. And what this method does, it simply measures the molecular brightness of GP6 labeled EGFP here in a small region of the membrane. And if there's two GP6s, then one. And what we have to have here are very important controls. This is a monomeric protein, CD86. CD28 is a dimeric because it's covalently linked. And you'll see that this is different from CD86. You'll see some very big bright spots up here, but that's because you've got two dimers coming together in the membrane at the same time. Put in glensosumab, and now it's preventing dimerization. So suddenly we're showing a new way of inhibiting activation, not just by steric hindrance, but also by preventing dimerization. Glensosumab blocks many more of the GP6 ligands, including fibrin, than nanobody 2. Does that make it a better inhibitor or not? That's a different question and very hard to speculate. But certainly it's got a different mechanism it's got an additional mechanism of action. Well, we've gone on to raise nanobodies to all of these 4 receptors, and again, always through VIB. You'll see that we were not raised, we only had 2 classes for pair 1, but then we only sent off a very small region of the protein because we wanted to go the heparin binding domain, because that's where we wanted the affinity ligands. Now, I've showed you that these nanobodies can be inhibitory, but we also wanted to use them to make ligands. So we thought, well, how many epitopes do we need to activate a receptor? Do we need a divalent, trivalent, tetravalent? We've made this with an FC domain, a mouse FC domain that does not bind to FC gamma R2A. We've also made them as linear tetravalent ligands, but I'm going to show you one results with this for reasons that I don't really have time to explain. But they both work very, very well. Firstly, what happens as you start to...
We are slowly running into the next session. So, Steve, if you could wrap up, that would be very appreciated. Thank you.
Absolutely. So if you make, what you can see is you make the length, you can increase their affinity. And now whereas the divalent ligand is inhibitory. The tetravalent ligand is an agonist. It's a potent agonist. It's more potent than those previous agonists.
Thank you. Thank you very much, Steve.
Yeah.
It's a very good overview of development in the area where we already have a lot of therapies. I think it would be best if it's possible for you to stay, and we will have a common discussion at the end because we need to move to the next session. There is one question in the chat, and we will forward it by email to you as well. I also have a question, but I will also send it to you. With this, thank you very much for these excellent presentations, and to both of you, and I will pass the chair to Pasquale.
Thank you, Tom, and thanks again, Steve, for the wonderful talk and accepting the invitation. We can move to the last session. What we will do now, we will skip the break because we are slightly delayed. And Alberico, as far as I understand, you have a plane waiting for you, if I'm right. So we move forward directly to Alberico Catapano, which is a professor of pharmacology. Alberico is a world-class leader in the field of lipid targeting therapies, in charge of several guidelines on behalf of the European Atherosclerosis Society and the European Society of Cardiology. So Alberico, thank you very much for accepting the invitation, and we are looking forward to your talk.
Thank you. Thank you very much. Can you hear me? So thank you very much, and apologies, the plane is not waiting for me, otherwise I would longer. Now, today I will talk about this demanding title, plasma lipid lowering, are we done? These are my disclosures related to this topic. Now, essentially, if one looks at the data we have collected over time and the role of LDL and ApoB-containing lipoproteins in causality, one could think that we have enough drugs there to be able to decrease the risk and reduce the rate of events. This has been related in the guidelines to the causality of LDL, which is based on genomics, genetics, epidemiology, pathology, intervention trials, you name it, and to the concept that risk is modulated according to the level of total risk in patients. In other words, lipoproteins provide you a higher risk in terms, absolute terms, if you are at higher global risk. Added to this, there's a concept that the more you lower the LDL, the better you are off, and this is largely independent on the type of drug you're using. Therefore, if you were to use a drug that is reducing LDL, it doesn't really matter because it is really something that works. Okay, the other thing we have learned with the guidelines is we are essentially good at reducing risk by lowering the LDL at any condition. Will that be the starting point for LDL? The relative risk reduction is really independent of where you start, the age, sex, the previous history of cardiovascular disease, diabetes, you name it. So what is the concept is, it is the relative risk reduction is independent of your clinical condition. What it matters really is the absolute risk condition where you stand, then that will make the difference in terms of the drug, in terms of reducing the number needed to treat, which is obviously the absolute risk reduction related. The other point we have learned over time with cholesterol, and this is true for LDL in any way you reduce it, and we'll see that in a minute, is it takes time to get the benefit. The first year, the benefit is not in full per millimole LDL reduction. The benefit comes with time and then stays roughly constant. This is not exactly so, and we will soon publish a paper that shows there is accrual of that, and this is important. As you can see here, this problem coming from the CTT meta-analysis, whereby the absolute risk was the risk reduction was calculated according to the level of LDL reduction on the first year of the trial, which was not exactly the same every other year. So you have to correct that, and was not done. In the end, the concept is the more you reduce the LDL, the higher the number of events you can spare in terms of the number of events, cardiovascular events spared per 10,000 people treated. Obviously, this is also related to the level of risk. The higher the risk, the more you are efficient there in terms of reducing events. You spare 1,440 events in 10,000 people treated with very high risk and 2 millimole reduction. That means a number needed to treat of 7. Exactly the same approach in a lower risk population will give you a number needed to treat of roughly 20. That makes the difference and makes also the results of a trial more interesting or less interesting to the people. We also have set in the guidelines and according to other documents what kind of drugs we should use. We have a number of very effective drugs nowadays at reducing LDL cholesterol, and this reduction is coming with statins, which is always recommended as a first-line therapy, but also ezetimibe, also the presence of PCSK9 inhibitors, and some other drugs that have become available, like bempedoic acid. Now, some people is suggesting, and we have done that as well in the guidelines, that you can start already, like what happens with blood pressure control, or even in diabetes in some instances, to reduce the LDL cholesterol straight away with a combination of 2 or even 3 drugs, depending on where you start from, because you have to reach the goal, and the more you reduce in absolute term LDL cholesterol, the larger will be the benefit. However, having said that, there is one point that still remains unresolved, and many others, obviously. One of them is the actual burden. This is an old slide from Orton from Dallas, indicating that depending on where you start with LDL, and this is the case of familial hypercholesterolemia. When you have a familial hypercholesterolemia, your LDL cholesterol by genetic causes is high, and build up your exposure to LDL cholesterol until you get to a point where the first evidence of cardiovascular disease is present. This is an exceedingly young age as compared to the average population without FH. However, we are able to change this trajectory if we were to treat and reduce LDL. The amount by which we reduce LDL and when we start, so the time of starting, is crucial though, because the slope of the curve will change, but if you change the slope of the curve earlier, then you intersect this line here, where is the hypothetical threshold for cardiovascular disease, evidence of cardiovascular disease, is much close to that of the normal population. Is this something that can be somehow proven? Well, genetics help us in that. If you look at randomized clinical trials, this is data we have published with Ryan here a few years ago in the LDL consensus. In a 1 millimole reduction for 5 years, these are reported to 5 years, gives you a 22 to 20%, 33% relative risk reduction. If you go with 1 millimole, but you go with the prospective cohort study, 12 years, the associated reduction is 33. And if you go by Mendelian randomization, 52 years of follow-up, 1 millimole LDL reduction will give you 53%. This is easily explainable by the burden, because in this way, for 52 years, you have had lower LDL cholesterol as compared to 5 years. The other side of the flip of the coin is that you can achieve the 22% by reducing LDL with moderate intervention, 0.3, 0.35 millimoles, that means 15 milligrams per deciliter, lifelong, and get a 22% relative risk reduction in the population. This contention of the exposure has been somehow validated in a follow-up study, an open label study with Fourier. This OLE study took with evolocumab, and patients who were in the placebo originally on the Fourier trial, 2.2, 2.3 years, were given therapy together with the ones who were already on evolocumab. They continued receiving the drug, the monoclonal antibody. What happened in these subjects? Well, if we look at the data about LDL cholesterol, obviously, when you start giving the drug, the PCSK9 antibody drops down, and you get here essentially the same as where the patients treated with evolocumab originally in the trial stood all the time. But the other important thing is the efficacy in terms of primary endpoint. If you look at the OLE open label study, then the people who was in placebo and moved to evolocumab had more events as compared to those who were in evolocumab and stayed in evolocumab for 5 years. And this is true also for the key secondary endpoint. And if you plot together the two lines, what you can immediately see is, this is obvious by eye inspection, after a period by which the placebo that moved to evolocumab continued to accrue events, then all of a sudden started to slow down, and the slope was exactly the same as in the people who were in evolocumab all time long. That's both the primary and secondary endpoints. If you plot that in a different way, the usual way we plot it, even in Fourier, you can see here the less effect in the first year as compared to the second year. And this is the lag time that is present for both primary and secondary endpoint. But then if you look afterwards, following up, what has been named by the author legacy, or you can call memory, you call whatever you wish. But this is essentially the fact that it takes time to go there. An obvious way of explaining that is the change in the burden that has been accumulated in the people taking already the drug, as compared to those who were first exposed to the drugs. Eventually, they will get to the same point. So this archetype here is demonstrated also with other drugs, and this has been shown over and over. And that appears to happen even when you have intervention on the top of other drugs. The other point is triglycerides. This is still an unsolved area in lipid metabolism and in pharmacological control. Observational epidemiology mistakenly indicated that there was no role because these data were corrected, adjusted for non-HDL. Non-HDL covariates with triglycerides, so correcting for itself obviously disappears. However, when you do Mendelian randomization, then things change. And you can have here a clear relationship between remnant cholesterol, which is, by the way, the triglyceride, because this is a measure of the cholesterol in VLDL and therefore directly related to triglyceride, as compared to what they say with LDL. If you do some studies, let's look at the right-hand side of the panel, where you do the Mendelian randomization, you can see the LDL cholesterol stays there, both with conventional or Egger analysis and Mendelian randomization. HDL disappears, so HDL is not proven to be a causal factor for atherosclerosis, and that's over and over the years. But then with triglyceride, they also stay per an increase of about 1 millimole, or standard deviation in this case. Now, if you put together two scores that are related to the LDL and the triglycerides, and you put this 2x2 analysis with the LDL receptor score on LPL score, what you can see here, both of them show a correlation. However, when you put them together, they go further down, indicating there is, as a minimum, the possibility of additive effect. And what happens if you look at one hallmark that is triglyceride and cholesterol, because the LDL and the LPL score reflect that. You can see here, they go up to 6.4. So this change here, LPL score only triglyceride, you change here reduction of LDL cholesterol, both of them together, even higher effect. So when you look at 10 milligram per deciliter, and you put them together, LPL score and LDL score perform exactly the same. The problem is to have this 10 milligram deciliter decrease in ApoB, which recapitulates the lipoprotein there. You need the 70 milligram per deciliter LDL reduction to cope with a 14 milligram per deciliter reduction of LDL. Again, if you plot everything by 10 milligrams, then everything fits on the same line, indicating, therefore, that reducing triglycerides will benefit, but you have to reduce them dramatically. And when you put all together triglyceride, LDL cholesterol, and ApoB in a multivariable Mendelian randomization analysis, then ApoB is the one that is coming up as the winner, if you will. In practice, what we have? Well, let's look at the triglyceride trials with the fibrates. These are before the trial with pimafibrate, the prominent. Altogether, in a meta-analysis, they appear to be effective. However, some of those trials, especially the more effective, were on a background without statin. If we go to the prominent study, which was run with pimafibrate, a specific fibrate that was designed to address only specific genes, fibrates are PPAR agonists, being similar in structure to fatty acids somehow. And you look for all the data changes, triglycerides were decreased. However, if you look at the decrease in terms of cholesterol, the change was quite little, even increased a bit. HDL increased a bit. LDL cholesterol was also increased. Further, if you look at the non-HDL, there was no change whatsoever in all the lipoprotein containing ApoB, including VLDL, albeit triglyceride went down. And apolipoprotein B level increased sharply. In the end, no effect. If you contrast that with the REDUCE-IT, this trial here, what the REDUCE-IT has shown us, this was icosapentatel, 4 grams per day. Very good results in terms of relative risk reduction and absolute risk reduction. And if you look at the data on the REDUCE-IT, on triglyceride, the reduction was 43 milligrams, it's roughly the same as in the previous trial. But LDL did not change in plus states with a reduction, but importantly also ApoB went down. The expected relative risk reduction, which is attributable to that, is in the order of 6 to 8%, which is not explaining the full effect of the icosapent, but that may explain why icosapent was effective, the reduction of ApoB. And if you compare the REDUCE-IT with another trial using another mixture of omega-3, HEPA, and HA, you can see here, triglycerides were reduced more or less to the same effect. However, in the REDUCE-IT, only the ApoB was decreased, not in the straight. Again, sustaining that. And if you meta-analyze, this is in our data not yet published, for ApoB reduction, you can put all the different interventions on the same line, both after 2 and after 4 years of treatment for those trials who have stood this length in the trial duration. Finally, another key point, in my view, is this one. This is again, from a study with alirocumab from the Odyssey. What they did in this trial, they eliminated the role of LDLs, so adjusted for the LDL reduction, and took the ApoB changes. So the ApoB changes corrected for the LDL reduction still predicted the event. So the lower went the ApoB, the better the results. So what about new drugs? Well, we've moved from the statin decades to second generation, to a new wave that is related to the genetics and genomics we have all along the line. What I would like for you to think, though, is, albeit here, we have a few targets that have been identified with the genetics. You can see here, angiopoietin-like 3, ApoC3, PCSK9, ApoC3. We have a number of pathways, more than 160 and growing, 163, that are related, or LOCI, that are related to CAD. And only a few are only lipids. Whether those are related to lipid metabolism and lipoprotein metabolism and depend to each other is still difficult to say. Yet, we have a long, long way to understand how this can be important in determining our capability of controlling lipid and risk accordingly. In other words, the vulnerability of a patient is still something we do not understand properly to when we stratify in each patient the risk according its susceptibility to given factor like increased lipoproteins or ApoB-containing lipoproteins. Here is another view of the type of drugs that have been and are in development and have been developed with these new approaches. So we have plenty of them coming. What will be their place in therapy? Well, to date, we have this RNA-based approach that some of them are already in practice, like inclisiran, which is a small interfering RNA that increases PCSK9 production in the liver. And we have essentially two approaches, antisense oligonucleotides or small interfering RNAs. Differences are substantial there. I'm not entering the mechanism, but it will suffice to say if we use this approach, it will take larger doses and more frequent administration as compared to this one, given the mechanism and given the way the drug is accumulating in the liver. And we have developed a number of new drugs, going from some small molecules, going to small interfering or azos. We have monoclonal antibodies now. We have antibodies that affect angiopoietin 3, for instance, in circulation. But what I can see for the future is in these three areas. CTP inhibition, there is a new wave of interest, not because of the HDL, which has been a failure, as probably would have been expected if data were looked upon carefully, but in the reduction of ApoB and LDL. This is an old, if you will, CTP inhibitor that decrease dramatically the LDL cholesterol. You can see here, 50%. And this is by preparative ultracentrifugation or by free rate calculation, which is quite more or less the same value obtained. And if you look at ApoB, 30% reduction. Non-HDL, 44%. So most probably this will be effective at reducing cardiovascular events because also reduces the burden and the number of particles. Lp(a) is another player. We know that Lp(a) is related to disease, and multivariable adjusted Mendelian randomization is related to the cardiovascular events. However, it's not normally distributed, and few people have a high level compared to a large number of people having relatively low level in the population. But Lp(a) plays an important role in determining risk, because if you look at the average level in the UK Biobank, which is this value, and you look at the baseline risk of atherosclerotic event here, and you were to calculate the risk of this population, and then the risk of the population having 150 milligrams per deciliter, 350 nanomole approximately, you can see here the jump in risk that is attributable only to the fact that is Lp(a) increased. So increasing that level of Lp(a) means there is an interaction with the risk, and the risk goes up quite dramatically. What drugs will decrease Lp(a)? PCS monoclonal antibodies do, and there are some hints that also in the trials, but this is statistically with manipulation, if you will, has been quite criticized. However, there is a reduction, which is minor though. Also, inclisiran, another PCSK9 inhibitor in a different way, decreases Lp(a). All of them go down about 20 to 25%. However, if you go with CTP inhibitors, obicetrapib, you can decrease by 56% by mechanisms that are completely unknown and need to be understood. But there are also azos, like pelacarsen, the third generation GalNAc, which drives directly to the hepatocytes, the azo. And you can see here the dramatic reduction. You can get up to 80–85% of Lp(a). And small interfering RNAs, like olpasiran, that has a reduction that is even stronger, 90%, and that lasts for longer, as expected, with small interfering RNAs. There is even another one that is not a name, but is SLN 360 that is even more effective and longer lasting, and is also smaller interfering RNA. So we have drugs, in our view, by which by reducing lipids, you reduce risk, and is related to Lp(a). And we have this vast array of approaches now, and this vast array of approach allows us to choose the proper drug for the proper patient, according to the baseline risk, according to what are the risks in terms of lipids. Would that be ApoB high, LDL normal? Would that be therefore high triglyceride, non-HDL cholesterol? Would that be Lp(a)? Would that be anything? However, there is one point that need to face, and we are not good at all at using the drugs we already have. We are using moderate intensity statin and low intensity statin in people who has established atherosclerotic cardiovascular disease. And this is a major mistake. In addition to that, we have a problem of adherence and compliance, which is dramatically low also for lipid lowering therapies in the long run. So in conclusion, we have multiple pathways for reducing the atherogenic lipoproteins, LDL cholesterol, ApoB at all in all, or non-HDL. We have combination therapies, and some new one will come that will allow us to address unknown areas to date, like Lp(a). Any KIT pathway can be targeted, including previously undruggable targets like Lp(a). Lp(a) is the most promising. Triglyceride, how? As I suggested here, I think looking for ApoB will be the best way, forgetting about triglyceride itself. Addressing the cholesterol ApoB burden will be a major challenge. That should be calculated, because if we were to change from our centralistic view of just one shot, we see at one point the LDL cholesterol, but we don't know what happened before, we can make big mistakes in evaluating how important is a given level of LDL or ApoB-containing lipoprotein in that patient. There are efforts to do that. Obviously, you require multiple determination. And then adherence and persistence to therapy, which is always a big problem and still stays. Thank you very much for your attention.
Alberico, thank you very much for the very comprehensive and in-depth overview, as usual. Do you have time to answer a couple of questions? Okay, fantastic. So can I start with the first one? We have been discussing about what we are trying to do today, and it's the establishment of a guide to cardiovascular pharmacology. And today, in the morning, we've already discussed how a guide may help to address some of the issues that you've highlighted in your talk. So, for example, there are 163 targets in GWAS, but we are only targeting few molecules. So, you are a pharmacologist. How do you think a guide can help? What the guide is not doing at the moment and may do with the help of the cardiovascular community, and how we can push things forward altogether?
I think it's almost impossible to answer your question first of all, Pasquale. However, having said that, I think it's important to realize we will need to be able to take into account all these different propensities of people to become susceptible to the disease. In other words, we are individual, we are not like each other, and each one of us will be more or less sensitive to a given proven causal factor for cardiovascular disease. This will be valid for LDL, will be valid for any other thing. And what the information that is coming from this GWAS, but also from other analysis, exome analysis would be probably much better, is informing us about the status of that patient, and we should be able to collect this information and make for each individual his particular status in terms of susceptibility to the disease. If we were able to do that, then we'll make a big step forward in order also to adjust therapy, intensity to the therapy, as well as starting early in treatment. I'm not sure that the way we are approaching now in terms of pharmacology, in terms of therapy, being, so smashing, I'm talking for lipids, is the right way. We put, we try to fill the holes, but the holes are already there. we can avoid that. It's been demonstrated, at least for cholesterol, for LDL cholesterol, but this is also true for blood pressure somehow. And we should be doing that. And also in terms of health. It's against pharmacology, if you will, because you can do that also by lifestyle changes. But I think this is an important thing. And there are strong arguments now for early intervention, not to very high intensity.
Okay, fantastic. Do we have any question from the panel?
You are mute, Carlo.
Carlo, you're mute.
How early would you start, Alberico?
Sorry?
How early?
How early you start?
How early would you start? Because it has political implications as well.
Obviously. I mean, I don't think that everyone will have to start a therapy. If start, you mean therapy. Changing lifestyle is not perceived as a therapy, albeit everything we do is a therapy in the end. But having said that, I will implement a large scale changes in lifestyle and approaches, keeping these risk factors low. At the same time, having the peak of the susceptibility, I will start as early as possible. As early as possible, I mean around the 20s, if you will be able to. And if needed, even with a low dose statin or even higher. And then you need the checkpoint. And then some of these people, maybe some, the presence of imaging of lesions you can detect in the vasculature. And that would be even suggesting for more intensive therapy if they proceed further.
Thank you.
I think that the answer in terms of the timing is extremely important, even in terms of global health. Alberico, as you know, I do a lot of work with Africa, and the mean age in Africa is 19.7, in Europe is 44.4, and we got in Nigeria, for example, 50% of people dying of cardiovascular disorders. So how early is in the global landscape is also a very interesting point, because clearly there is a very young population that is struggling worldwide.
Yes.
Alberico, thank you very much. I know that you need to leave. You cannot join us for the discussion, but we will be in touch because your contribution will be fundamental.
Thank you. Thank you very much. Apologies for
You're
being
welcome.
In a rush.
And now we can move forward to the last speaker, which is Professor Carlo Patrono. Carlo, are you able to share now? Yes, fantastic. So Carlo is a professor again of pharmacology from the Catholic University of Sacro Cuore in Rome, and he has been working all of his life on targeting cyclo-oxygenase in cardiovascular disorder. So thank you very much again for contributing, and the floor is yours.
Thank you, Pasquale, for this kind introduction. Oops. Cannot advance my slide. Okay. So this is my disclosure slide. And these are the cyclo-oxygenase isozymes, which, as you know, are largely ubiquitous enzymes that catalyze the first committed step in prostanoid biosynthesis. Now, aspirin is a prototypic cyclo-oxygenase inhibitor, although it has some unique features whereby it tends to selectively inhibit beta-COX-1 at low doses, and then it inhibits COX-2 in a dose-dependent fashion when it's used as an analgesic anti-inflammatory agent. The unique features of the mechanism of action of aspirin in inactivating cyclo-oxygenase are illustrated in this three-dimensional structure of the enzyme, and in particular the so-called cyclo-oxygenase channel, whereby aspirin first binds reversibly to this common docking site for all non-steroidal anti-inflammatory drugs, and then selectively acetylates this serine residue at position 529. And the key to this mechanism of action is so-called hit and run. So it permanently blocks the cyclo-oxygenase channel near the catalytic pocket of the enzyme, and then runs because it has a very short half-life. And this is ideally suited to inactivate COX-1 in platelets because platelets do not have the capacity to resynthesize the enzyme during the 24-hour dosing interval. And these features result in cumulative and biochemically selective inhibition of platelet COX-1 as compared to renal COX-2, which is substantially spared during the administration of a dose as low as 30 milligram daily, which represents 1/10 of a single analgesic dose and about 1% of a typical daily dose as an anti-inflammatory agent. And this is largely due to the fact that by virtue of this unique mechanism of action, aspirin induces cumulative inhibition of the enzyme in anucleate platelets, while nucleated cells, for example, of the kidney, have the capacity to resynthesize whatever enzyme has been acetylated by aspirin within the 24-hour dosing interval. And this in turn results in substantial reduction in major vascular events in high-risk patients. This is a meta-analysis by Colin Bagent and the antithrombotic trialist collaboration, showing that in trials of doses as low as 75 milligram daily, there is a 1/3 reduction in major vascular events. And trials using twice as much or 10 times, 20 times as much aspirin, there is no evidence of an increase in efficacy. If anything, there is a slight inverse relationship between the daily dose and the clinical efficacy of the agent. And these findings have two important messages. One is that the clinical benefit of aspirin as an antithrombotic agent is saturable at low doses, exactly like acetylation of the enzyme is saturable, and like inhibition of thromboxane biosynthesis is saturable at similar low doses. And the second message is that once you have profound and complete suppression and long-lasting suppression of platelet COX-1 activity, adding COX-2 inhibition in a dose-dependent fashion by increasing the daily dose of aspirin does not seem to add anything in terms of potential benefit. And if anything, the opposite may be true, perhaps due to dose-dependent inhibition of prostacyclin production. Now, the data I showed you come from indirect comparisons with all the limitations of indirect comparisons, but there is at least a very large head-to-head comparison of low-dose aspirin versus intermediate-dose aspirin, 300 to 325, in over 25,000 patients with acute coronary syndromes, the current OASIS 7 trial. And in terms of the 30-day hazard of the primary endpoint of death from cardiovascular causes, myocardial infarction, or stroke, there is no substantial difference between a lower dose and a higher dose. As you know, throughout the years, there have been proponents of non-cyclo-oxygenase dose-dependent effects of aspirin. Well, this sort of evidence doesn't really support the importance of these non-cyclo-oxygenase dose-dependent effects of aspirin. What about reversible inhibitors of the same target? Well, the only reversible COX inhibitors that have been tested in randomized clinical trials for their antithrombotic efficacy are sulfentrazone, indobufen, flurbiprofen, and trifluzel. However, none of these reversible inhibitors is approved as an antiplatelet drug by the FDA, and it's really unclear under which circumstances they may be prescribed instead of aspirin in other countries. As you know, reactive oxygen species that are generated as a consequence of a variety of metabolic disorders can catalyze the peroxidation of esterified arachidonic acid to form a family of prostaglandin isomers called isoprostanoids or isoeicosanoids, some of which have interesting biological activities like isoprostaglandin F2 alpha, which is an agonist of the thromboxane receptor on platelets. So some 20 years ago, there was a pretty solid rationale for expecting that a thromboxane TP antagonist might show clinical superiority as an antithrombotic agent vis-a-vis low-dose aspirin, in as much as low-dose aspirin would only block platelet COX-1-derived thromboxane, while a TP antagonist would block the action of all sources of thromboxane, including inflammatory cells expressing COX-2, and in addition would also block the agonist activity of some F2 isoprostanes. So there is at least one thromboxane receptor antagonist that has undergone phase 3 clinical development. This drug was developed by Servier. It's called terutroban. And the very large phase 3 trial called PERFORM in over 19,000 patients with a recent stroke or TIA compare head-to-head low-dose aspirin and the TP antagonist terutroban. This is the Kaplan-Meier curve for the proportion of patients with reaching a primary composite endpoint, the usual triple composite endpoint, non-fatal MI, non-fatal stroke, vascular death. And this is what happened in aspirin-treated patients. This is what happened in terutroban-treated patients. Not much of a difference. In fact, as the ratio was 1.02. These are the number of events. So with about 2,000 events in the trial, there is very robust statistical power to exclude a major difference between the two, and the 95% confidence interval are compatible only with a minor difference one way or the other. So unfortunately, no other thromboxane receptor antagonist has undergone comparable clinical development in other clinical areas. So for the time being, we can say that the role of aspirin insensitive agonists of the thromboxane receptor seems to be quite limited in patients with cerebral vascular disease. Whether this is also true for coronary ischemic heart disease, it's an open question. What about safety? Well, it's interesting that in the same PERFORM trial of, as I say, over 19,000 patients, all bleedings were substantially the same. If anything, those associated with aspirin were numerically lower than those associated with terutroban. And most interestingly, the risk of developing gastrointestinal bleeding was quite comparable in the two treatment arms, a finding which is consistent with the view that gastrointestinal bleeding in patients treated with low-dose aspirin is essentially related to its antiplatelet effect. Now, as you know, aspirin is considered by many intrinsically gastrotoxic because of an unfavorable heritage of its use as an analgesic anti-inflammatory drug at much higher doses. But if you look at the results of this rather large endoscopy study in over 1,100 patients with osteoarthritis who were randomized to receive placebo, aspirin 81 milligram daily, or ibuprofen 800 milligrams 3 times daily. Low-dose aspirin does not produce more gastroduodenal ulcers than a placebo, in contrast to ibuprofen, which increases the risk of developing gastroduodenal ulcers by 3 to 4-fold. This is a 12-week study. What about long-term? Well, there is a recent study published last year in JACC, where they use magnetically controlled capsule endoscopy for the assessment of antiplatelet therapy-induced gastrointestinal injury throughout the gastrointestinal tract. And this is the rate of gastric or small intestinal ulceration at 12 months in patients, acute coronary syndrome patients who were treated in the long term with either aspirin, clopidogrel, or dual antiplatelet therapy. And as you can see, the rate of this ulceration in aspirin-treated patients is also numerically lower than that associated with clopidogrel. We heard already from Steve Watson that there is more to platelets, to activated platelets, than hemostasis. And this slide illustrates the fact that activated platelets may participate in the early phase of colorectal carcinogenesis. This is based on a number of placebo-controlled low-dose aspirin trials in patients undergoing colonoscopy for colorectal cancer screening, showing a reduction in the risk of developing in recurrence of a sporadic colorectal adenoma to a comparable extent as that associated with traditional NSAIDs and COXIBs, suggesting that perhaps products released by activated platelets at sites of intestinal mucosal injury are involved in COX-2 induction and prostaglandin E2 formation and the downstream events that may contribute to colorectal carcinogenesis. Now, I mentioned COXIBs, and as you know, one of the major COXIBs, rofecoxib, was withdrawn from the market because of the gastrotoxicity unveiled during one such study for the prevention of sporadic colorectal adenoma recurrence. So this slide summarizes the 5 different molecules that completed phase 3 clinical development. All of them reached the market in some countries. The 2 compounds developed by Merck, rofecoxib and etoricoxib, the 2 developed by Pfizer, celecoxib and valdecoxib, and lumiracoxib developed by Novartis. And if we were to put a parent compound up here, we would have to place diclofenac, which is structurally related to lumiracoxib. Now, 3 of these molecules have been withdrawn from the market because of various reasons, beginning with rofecoxib withdrawn in 2004 because of the emergence of increased risk of cardiovascular events, atherothrombotic events, particularly coronary events. Together with Colin Bagent and many other colleagues, we established a COXIB and traditional NSAID trialist collaboration. And it took us some 7 to 8 years to collect individual participant data from all the COXIB and traditional NSAID trials to look at vascular and upper gastrointestinal effects of cyclo-oxygenase inhibitors, essentially COX-2 inhibitors. And this slide summarizes the effect of COXIBs on major vascular events. COXIBs increase the risk by about 40%, largely due to roughly a doubling of the risk of fatal and non-fatal coronary events, and a statistically uncertain increase in cerebrovascular events. Now, of course, COX-2 selectivity is a continuous variable, even though talking about nomenclature and semantics, the many people have used this dichotomous definition of selective and non-selective COX-2 inhibitors, which is really not supported by COX-2 selectivity being a continuous variable with a large overlap between some traditional NSAIDs and some first-generation COX-2. Well, it was thought that cardiovascular hazard was a function of increasing COX-2 selectivity, because the first drug to be hit by this problem and to be withdrawn from the market was rofecoxib, a highly selective COX-2 inhibitor. But in fact, in our analysis of all the celecoxib and rofecoxib trials, and here you see the number of trials, the rate ratio for major vascular events is substantially identical, suggesting that this is a mechanism-based class effect of all these agents. What about the traditional NSAIDs? Well, interestingly, some time ago, we had shown that naproxen, when given at a high dose of 500 milligrams twice daily, mimics the antiplatelet effect of low-dose aspirin in suppressing thromboxane biosynthesis in vivo, as reflected by the urinary excretion of this thromboxane metabolite, to a comparable extent as low-dose aspirin over the 24-hour dosing interval. And so we pre-specified a separate analysis of naproxen from non-naproxen traditional NSAIDs, largely ibuprofen and diclofenac, in this individual participant data meta-analysis. And as you can see, naproxen is associated, and this is high-dose naproxen, is associated with a neutral cardiovascular phenotype, in contrast to high-dose ibuprofen and high-dose diclofenac that recapitulate the same 40% increase in risk of major vascular events as shown in the COX-2 trials. Now, based on some of these promising findings, a company called Nycox developed a number of NO NSAIDs and concentrated on naproxenol, which was a novel cyclo-oxygenase inhibiting nitric oxide donator compound called Synod, which was developed to reproduce the same analgesic anti-inflammatory effect of a traditional NSAID, with a potential advantage related to NO release, potentially contributing to improved gastrointestinal safety, but also increased vascular safety vis-a-vis traditional NSAID by virtue of NO donation and the expected vasodilator effects. This naproxenol has undergone clinical development up to phase 3, and these are the results of a phase 3 trial, a 53-week prospective randomized multicenter trial in patients with osteoarthritis, where 2 doses of naproxenol, 375 and 750, were compared to naproxen 500 milligrams twice daily and to placebo. And we're looking at data from a pain subscale or a function subscale of or a patient's overall rating of disease status. The 2 doses of naproxenol were more effective than placebo, shown with these gray triangles. And the higher dose of naproxenol was comparable to the equimolar naproxen dose of 500 milligrams twice daily. So the drug was able to replicate the clinical efficacy as an analgesic and anti-inflammatory agent. And so it was established as being non-inferior to naproxen 500 milligrams twice daily. And then they also assessed its effect on blood pressure in the same type of patient population, patients with elderly patients with osteoarthritis, and found that in contrast to naproxen 500 milligrams twice daily, that as compared to placebo, increased blood pressure by about 5 millimeter mercury of systolic blood pressure. Naproxenol at the equimolar and comparable clinical efficacy dose of 750 milligrams twice daily did not differ from a placebo. So meeting the expectation that the drug would be potentially safer from the vascular point of view. But of course, there is more to increase probability of coronary atherothrombosis than the increase in blood pressure, because substantially you have COX-2 inhibition in different locations, vascular endothelial smooth muscle cells, resulting in reduced prostacyclin biosynthesis and unrestrained platelet activation onto ruptured coronary plaques. You have inhibition of COX-2 in cardiomyocytes, and then inhibition of COX-2 in the renal, cortical, and medullary structure, and improving the effect of an agent on blood pressure would only partially attenuate a potential cardiotoxicity. In any event, the FDA requested a cardiovascular outcome study to potentially approve this new compound, and the company decided to discontinue its further development. So we are left with the uncertainty about its potential long-term cardiovascular safety. Now, if we go back to the cyclo-oxygenase pathway of arachidonic acid metabolism, one potential alternative to targeting cyclo-oxygenase would be targeting the downstream enzyme converting the prostaglandin endoperoxides into prostaglandin E2 and its interaction with the EP receptors. Now, interest has been focused on a microsomal inducible enzyme called MPG synthase 1, which was cloned and characterized some 25 years ago at the Karolinska instituted by Bengt Samuelsson group, in particular by Per Johan Jakobsen. And Per Johan has co-authored a recent review on MPG synthase 1 inhibitors from preclinical studies to clinical applications. And his conclusions are quoted in this summary slide. 20 years have passed since the discovery of human MPG synthase 1, and there are no approved inhibitors for clinical practice. Although potent and selective human MPGS1 inhibitors have been developed and evaluated in clinical settings. However, there is sufficient heterogeneity in the structure between the human and rodent enzyme, challenging the possibility of developing potent inhibitors that function in preclinical models and can serve as clinical candidates. And given that all the NSAIDs are black box labeled and advised to be used with caution by the FDA, there is a guilt by association to target the prostanoid pathway with MPGS1 inhibitors. I'm not aware of any ongoing clinical development program with such compounds. I know that Merck, AstraZeneca, and Eli Lilly, who were among the first to develop promising inhibitors of this target, have discontinued these programs. To what extent this is due to difficulty in obtaining ideal clinical candidates vis-a-vis the requirement by the FDA for a cardiovascular outcome study is unclear at the moment. So to conclude, COX-1 is an unsurpassed platelet target to prevent atherothrombosis, and platelet COX-1 is a promising target to prevent colorectal carcinogenesis. And I believe that in thinking at a cardiovascular portal, we should also think about crossing across different therapeutic areas, a theme that was briefly touched upon by Ziad Malat this morning. Targeting platelet thromboxane synthase or TP receptor has failed to meet the expectations, the superiority expectations vis-a-vis low-dose aspirin, though within the limits of phase 3 clinical programs, which to my knowledge are available only for one such compound, that is tirutrubant. Targeting COX-2 in coronary inflammation or colorectal carcinogenesis is restrained by concomitant PGI2 suppression, and naproxen showed interesting results, but its further development would have required a cardiovascular outcome study. And finally, MPGS1 is a promising target to overcome the cardiovascular hazard of COX-2 inhibitors, but the clinical promise of these inhibitors remains unfulfilled. Thank you very much for your attention.
Carlo, thank you very much for the very nice and comprehensive overview, and also probably is the best way to finish this day, explaining how targeting the arachidonic acid cascade is complicated, and we still have a lot to do in order to reach to the best target and in the best manner possible. I think we are 10 minutes late, and I know that Amrita, we have a close discussion afterwards. So probably if there is any burning question from the panel, I think there is no one in chat. Otherwise, I will progress to closing the meeting. Do we have any question, Amrita?
I can save my questions for Carlo in the closed session.
Perfect. So I would say, Carlo, thank you very much for the contribution. Can I just remind everyone that we will be meeting with the speakers and the British Heart Foundation in 10 minutes, basically. And I thank you all for attending the day. I hope it was useful for all of you. It was definitely for me. We have been hearing a lot of different therapeutic targeting cardiovascular disease, and I've learned a lot. Amrita, is there any closing remarks that you would like to make before the ending of the meeting?
Just to say that I think it's been enormously informative. It shows us that the way to work out where the best value comes in these information resources is to make sure that one has discovery scientists talking with clinician researchers too at the same time, because our hope is that we generate something that's useful for the entire community. And clearly the way that a clinician researcher might enter the guide is going to be slightly different from the way a discovery scientist would enter the guide to pharmacology. Side effects, organs, cell-to-cell interactions. There are lots of different ways in which we need to think about in terms of determining how the resource might be useful for the wider community. The talks have been fantastic. Thank you, all of the speakers, for your contributions. Very grateful that you've taken the time to speak with us. We will be sending recording to all those who've registered for the meeting, and we will address any questions that were unanswered by those who attended the conference. Thank you everybody again, and we will be in touch, and I look forward to seeing all of the speakers in 10 minutes time together with the British Heart Foundation.
Thank you all. Bye guys.